So, hello everyone. After a successful completion of the one short series of this particular chapter, which is the breathing and exchange, I have brought another chapter for you, which is biomolecules. So, sir, this biomolecules is a chapter which we feel very hard sometimes, but it's not a hard chapter. It's a very easy chapter, and from this biomolecules, lots and lots of questions comes. So today we are going to try to complete this biomolecules within 2 hours, and I'll make sure that you will be able to solve each and every questions of the biomolecules through this series. I hope that each and every one of you are ready for this session, and I am live. Great, bachcho, great. So let's quickly start the session. Hi Bhavik, how are you? Good to see you. Hi music. Hello. So today we are going to check this biomolecules, right? Chalo. So let's quickly start the session, bachcho. So if I talk about the biomolecules, what is biomolecule? What do you understand by biomolecules? So biomolecules are those kind of substances which is present in the living organism. This is the for sure. If we are talking about the biomolecules, that means those molecules should be present in the living organism. Yes or no? Yes. But there are some confusions, sir. If I am gonna talk about the carbon dioxide, sir, carbon dioxide is also present in our body. But can we say that this carbon dioxide is a biomolecule? No, bachcho, no. Carbon dioxide is not a biomolecule. Why? Because the definition of biomolecule says that all the organic component, organic carbon containing, all the organic carbon containing molecules are known as what? Are known as biomolecules, which are present in the living organism. Is it clear to every one of you? Is this clear? Is the definition of the biomolecules clear to everyone? Yes. I hope that it is clear. Yes or no? Chalo. Great. Now moving towards the next examples. If I give you the example, let's suppose that carbon dioxide, to sir, we saw that this carbon dioxide is not a biomolecules. Why it is not a biomolecules? Because it is inorganic. It's not organic, guys. बच्चों ये inorganic है, organic नहीं है. Clear? That's why it is not a biomolecule. Now there are lots and lots of molecules which are present in our body, like potassium ions, calcium ions, sodium ions. All of them are not biomolecules. So sir, what is biomolecule? The question is, what is biomolecules? If I talk about the examples of the biomolecules, we can have proteins. Yes or no? We can have proteins. Guys, we need to complete this chapter today, okay? Everyone, we are gonna complete this chapter no matter what, okay? Proteins. Yes. Now what? We have carbohydrates. Next we have what? Nucleic acids. So these are what? These are the biomolecules. Amino acids, lipids. Yes or no? Yes, everyone? Great. Chalo. Let's move on now. We have discussed the definition of the biomolecules. Now, sir, if I talk about the biomolecules, so biomolecules are further divided into two categories. Biomolecules are further divided into two categories. What categories, sir? One is the, if you have a big biomolecule, then you are going to call it as biomacromolecules. If you are having a big molecule, then you are going to call it as biomacromolecules. And if you are having small molecules, then we are going to call it as what? Then we are going to call it as biomicromolecules. But sir, can we just see and say that this is small molecule and this is big molecule? Is that so? Can we just see and say that this is small, this is big? No. That sounds very, we can't have a proper judgment there. So let's give it a size thing. So if suppose it is, when we are measuring it, if it is showing weight of if it is showing weight of more than 1000 Dalton, more than 1000 Dalton, then it is known as biomacromolecules. Then it is known as biomacromolecules. And if it is showing the weight less than 1000 molecules, between 18 to 800 Dalton, it is known as what? It is known as biomicromolecules. Yes or no? Then it is known as biomicromolecules. Clear to everyone? Yes or no? Is it clear? Let me know in the comment section fast. Let me know in the comment section fast. Okay, chalo. Let's do the examples of biomacromolecules and biomicromolecules now. So see, if suppose you are having a chicken with you, or you are having any living tissue with you, right? Take any living tissue. Let's suppose you have taken a chicken. Let me draw a chicken here. Okay. Suppose that you have taken a chicken. Very good, Bhavik. How to draw a chicken? Is this a chicken? Let's suppose it's a chicken leg. Okay. It's a chicken leg. So what you need to do is that you need to bring mortar and pestle. You need to bring mortar and pestle. Keep this chicken in mortar and pestle and add some trichloroacetic acid. Add some trichloroacetic acid, right? Trichloroacetic acid. So what we have done? Taken a chicken liya हमने, and what we have added? We have added trichloroacetic acid. Now what, sir? Smash it, mix it. क्या करना है? मोटर और पिस्टल लेना है, चिकन डालना और उसको एकदम थिक स्लरी बना देना. Make a thick slurry. Make a thick slurry. Now what, sir? After this you have done, you have to take a channi. You have to take a channi or a cheesecloth. You take channi or a cheesecloth. Now what you are going to do is that you are going to take that slurry and keep it in the cheesecloth and squeeze it. So what will happen? Some of the things will be present in the cheesecloth, which will be known as what? Which will be known as what? Which will be known as retentate. Retentate. And some of the things will be what? Will be crossing the cheesecloth, and that will be known as filtrate. Yes or no? Filtrate. Clear this much? This much clear to everyone? Right? So sir, we have two things here. Now we have two things. 1 second, I'll just make it. Yes. So now we have two things. What, sir? One is retentate. Retentate. I'll be covering all the things, guys. This will be your last video of biomolecules. I'll try that you learn each and everything here only. So if you're gonna stick with me till 8 o'clock, I will finish your biomolecules. Maybe less than also, like we can complete it till 7:30 also, but maximum 8 o'clock, right? Okay, chalo, great. So we have what? We have retentate. We have retentate. So why retentate, sir? Retentate couldn't cross it, right? That means they are bigger molecules. So in retentate, we are having what? We have biomacromolecules. We have biomacromolecules. And if I talk about the filtrate, guys, and if I talk about the filtrate, so what is gonna happen in filtrate? Sir, in filtrate, the molecules were small. Yes or no? The molecules were small, and so that's why they are able to pass through it. So we can call it as biomicromolecules. Biomicromolecules. But sir, is it correct? Is it correct? Like we are saying that because of big molecule it remained in retentate, and because of small molecule it remained in the filtrate. This way so we can learn. But the main thing is what? The main thing is that the molecules which are present in retentate were not soluble in acid. This is what retentate contains non-soluble or acid insoluble substance. What it carries? A non-soluble, or I can say acid insoluble. That's why they remain in the retentate. And filtrate, whatever the things present in the filtrate is what? It is acid soluble. It is acid soluble. Yes, Shivesh, yes. That's the That's the thing I need here. That's the thing I need here, right? So it is what? It is acid soluble, right? Now let's see what molecules are acid soluble and what molecules are acid insoluble. Yes or no? So generally macromolecules are acid insoluble. Example is what? Example should be protein. Example should be protein. Example should be carbohydrates. Carbohydrates. And there should be some examples like lipids. Lipids. I'm making a star mark in lipids. We are going to discuss about the lipids afterwards, little bit afterwards. Okay. Now in biomicromolecules are what? Biomicromolecules, amino acids. See, if I talk about protein is made up of what, sir? So you are going to say protein is made up of amino acids. Yes or no? Yes. So the amino acid is basically what? Amino acid is basically the smallest part of the protein. So amino acids will come under the biomicromolecules. In a similar manner, let's suppose I am talking about the starch. Starch is a carbohydrate. And if I ask you that carbohydrate is made up of what or starch is made up of what, you are going to say glucose. So the smaller unit of any big substance is present in the biomicromolecules, and the bigger whole chain is present where? Is present in the biomicromolecules. I hope that this thing is clear to everyone. Yes or no? I hope that this thing is clear to everyone. Yes or no? Yes? Great. But sir, lipid is what? Lipid is something different. Lipid is a very badmash person. Lipid is what? Lipid is very badmash person. Lipid is technically what? It is biomicromolecules. Lipid is what? It is biomicromolecules. But why, sir? Why it is present in? Why it is present in acid insoluble pool? Anyone can answer? Can you answer? Can anyone answer? Can anyone answer why it is present in biomicromolecules? So see here. First of all, what we did is that where is lipid present? Lipid is the constituent of the cell membrane. Yes or no, बच्चों? Lipid कहाँ present है हमारे body में? Sir, it is present in the cell membrane, right? Now, when you have made the slurry, the cell membrane broke. Let's suppose this was the cell membrane. This was the cell membrane, and we broke this cell membrane in very small particles, right? Now, if I talk about the lipid, so lipid has two things. Lipid has two things. One is what? One is the head, and the other one is what? Other one is the, one second, I have written the spelling of head wrong. Are you serious, sir? What are you doing? Okay, leave it. Ignore the spellings. We can ignore the spellings, right? So, there is head, and this is what? This is tail. Now, among this head and tail, one is what? One is hydrophilic, and the other one is what? The other one is hydrophobic. Yes or no? So, when we break it, what happens? They form vesicles. These small part of cell membrane, they round up and they forms vesicles because their tail is hydrophobic and it doesn't want to get connected with the water. So that's why they make a surface. And when they are making the vesicles, their what? Their weight is increased and they are not able to, sorry, when they are making the vesicles, they are not able to pass through the cheesecloth. Their size is increased. So that's why we are able to find lipid in the biomacromolecules part. But if I ask what is lipid actually, so actually the lipid is what? The lipid is biomicromolecules. Clear to everyone? Clear to everyone? Yes or no? I hope that this thing is clear, right? I hope that this thing is clear to each and every one of you. Chalo, great. If this is clear, let's move on. Let's move on and let's talk something about the proteins. Let us talk about the very first molecule, very first biomolecule, which is protein. Ready everyone? Hi Sudarshan, how are you? Let us talk about the very first molecule, which is protein. Okay, just let me write one second, bachcho. Okay, chalo, chalo. So if I talk about the protein, what is the smallest part of the protein? From which part the protein is made up of? From which part the protein is made up of? So sir, the protein is made up of very small amino acids. Yes or no? The proteins are made up of very small amino acids. Now my question is, what is the amount of amino acid present in the living organism? What is the amount of amino acids, number of amino acids present in the living organism? So the number of amino acids present in the living organism is 20. Yes guys, only 20 amino acids are present and they are only making huge number of proteins, right? So what is amino acids? Amino acids are a monomer of protein, and when lots and lots of amino acids comes and then joins, then we form a protein. Now this amino acids are what? This amino acids are joined with a bond which is known as, is it glycosidic bond or is it peptide bond? Yes or no? Let me know in the comment section fast. Two amino acids are joined to form a glycosidic bond or a peptide bond. Yes, 20 pieces. Which type of bond? So sir, this bond is known as peptide bond. This bond is known as peptide bond, bachcho. Yes or no? This bond is known as peptide bond, right? Now one more thing which you have to make it clear, that exactly Bhavik, one more thing which you have to make it clear, that sir, protein is a heteropolymer or a homopolymer? Protein is a heteropolymer or homopolymer? Tell me. So it is what? It is heteropolymer. Why? Why we are saying that protein is heteropolymer? And I will say one more thing. Protein is always heteropolymer. Protein is what? Always heteropolymer. See, if you take any chain of the protein, it will be made up of what? It will be made up of different amino acids. No protein chain contains the chain of same amino acid. Like ऐसा नहीं होता glycine मिलके पूरा protein का chain बना दिया, नहीं. There are lots and lots of amino acids which are making a protein chain. So protein will be always heteropolymer. Yes or no? Is this clear? Yes or no? Great. Great, bachcho. Great. Yes, chalo. Good. Now let's talk about what? Let's talk about the amino acid. Let's talk about the amino acid. We need to know, na, we saw that protein is made up of amino acids. So to check it, we should know the structure of amino acid, right? So let me tell you about the structure of amino acids. Structure of amino acids. We will do a poll session tomorrow on this topic, right? Bhavik, music, Shivesh, Sudarshan, we will do a poll session on this topic tomorrow, right? Chalo. Now let's talk about the amino acids. So see, have you all seen methane? I know my chemistry is not very strong, but have you all seen methane? Methane तो मुझे बनाने आता है यार बच्चों, ऐसे मत बोलो. हाँ, methane तो मैं बना ही लूँगा. क्या होता है methane? Methane is CH4. Yes or no? Methane is CH4. So I'm gonna make methane like this. I know this structure is chemical. According to chemical bonding, this is not correct. According to chemical bonding, I should make this structure. Yes or no? See, I know little bit chemistry. Chalo. So according to chemical bonding, this will be the correct structure. Yes or no? But I'm making like this. I am making like this. So see here. See here, bachcho. So amino acids are basically what? It is a kind of methane, and from methane what you do is that you start removing 1,1H. What we have to do is that we'll start removing 1,1H. So let's remove H from this side first. 1 second, why my eraser is not working? Huh? Batao. Let me know. 1 second. Tetrahedral. Yes, tetrahedral. ये क्या हुआ बच्चों? चलो आ गया. So let's remove acid from here. Let's remove hydrogen from here. And when I am gonna remove one hydrogen, so in place of this, what I am gonna keep? I am gonna keep COOH, which is what? What do you call it as? What do you call it as? Alcohol? No. This is what? This is carboxylic acid, right? Now what I have done is that I have removed this H also. I will be removing this H also. Sare ke sare H I will be removing. So I remove this H, and in place of this, what I have added? I have added an amine group NH3. I have added what? NH2. I have added an amine group, right? Now, अब एक हाइड्रोजन को छोड़ देंगे, तरस आ गया हम पे, तरस आ गया इन पे कि चलो भाई एक हाइड्रोजन छोड़ देते हैं. तो ऊपर वाला हाइड्रोजन क्या करा हमने? ऊपर वाला हाइड्रोजन हमने छोड़ दिया और नीचे क्या लगा दिया? यस यस, अमाइन ग्रुप. नीचे क्या लगा दिया बच्चों? नीचे लगा दिया आर ग्रुप. नीचे क्या लगा दिया? आर ग्रुप, राइट? नीचे क्या लगा दिया? आर ग्रुप. Now what is this R group, sir? It can be C. It can be methane, it can be methane, it can be ethane, it can be benzene, whatever it can be. Okay, so this is what? This is the variable group. Like in amino acid, hydrogen COOH and NH2 will be fixed in all amino acid. The only group which will be variable will be this R group. Yes or no? Will be this R group, variable. Okay, will be this R group. Clear to everyone? Yes, Shivesh, very good. Is this thing clear to everyone? Let me know in the comment section, bachcho. It can vary, exactly. It can vary. Okay, chalo. This is clear. So that's why we are calling it as what? We are calling amino acids as substituted methane, because methane was there and we have removed what? We have removed their hydrogen. Yes or no? So substituted methane. What is amino acids? Amino acids are substituted methane. Clear to everyone? Great, bachcho, great. Now one more thing. See, if we talk about the structure of amino acids, so can you see that this is a chiral carbon, bachcho? Can you see that this is a chiral carbon? Yes, Bhavik. Can you see, is this a chiral carbon? Yes. What is chiral carbon, sir? A chiral carbon is that carbon in which all the 4 bonds are different. So yes, this is a chiral carbon, right? Okay. And this carbon is also known as alpha carbon. This carbon is also known as alpha carbon. Clear? This carbon is also known as alpha carbon. Is it clear to everyone? Yes? Okay. Now one more thing, I'm going to give you an example. Let's suppose that, let me know if this is a structure of protein or not. Sorry, amino acid or not. Is this the structure of amino acids or not? Can you say that this is the amino acid? Can you say that this is the amino acid? Anyone in the comment section, can you say that this is the amino acid? Sir, this is not the amino acid. Why, sir? Because the COOH group is not attached to the alpha carbon. The COOH group has to be attached to the alpha carbon. Then only we are going to say that it is an amino acid. Exactly, Bhavik. No? Is that so? What no? No, किसके लिए बोला भाविक? किसके लिए नो बोला? बताओ. इसके लिए प्रोटीन का स्ट्रक्चर के लिए कि मैंने कारिल कार्बन गलत बता दिया तुम्हें? थोड़ी बहुत आती है केमिस्ट्री, का गलत बता दिया तो माफ कर देना, बट मैंने सही बताया तुम लोगों को. चलो, ओके. नाउ सी, now what we have to do is that we have made what? We have made the this, what should I say? Amino acid. Okay, now we have made the amino acid, and after that what we have to do is that we have to make the proteins. So how we are going to make the proteins? We are going to make the proteins with the help of peptide bond. We are going to make the proteins with the help of peptide bonds, right? हंस लो तुम लोग. मैं एक फिजिक्स का क्वेश्चन दूंगा ना अभी, उमा शंकर सर को बुला के एक फिजिक्स का क्वेश्चन दिलवा दूं ना तुम लोगों को, फिर मैं ऐसे चुप के हंसूंगा. बुला दूं क्या उमा शंकर सर को? चलो देखो. So peptide bond, how we are gonna make it? So this was a protein. this was the this was the amino acid. We have CCO. Here we had the here we had what? Here we had, let's suppose, hydrogen, and let's suppose R group we have, and here we have the NH2. Yes or no? Here we have the NH2, right? शिवेश से पूछो, उसको बिल्कुल फिजिक्स नहीं आती है ना शिवेश, मेरी तरह, जैसे मुझे फिजिक्स नहीं आती, राइट? चलो सो सी हियर बच्चों, सी हियर। चलो अब आ जाते हैं वापस, है ना? तो यहाँ पे क्या है, यहाँ पे हमारे पास एक प्रोटीन का अमीनो एसिड का स्ट्रक्चर है। ये क्या है? COOH। ये क्या है? एक प्रोटीन का स्ट्रक्चर। सो फिर दूसरा नॉट प्रोटीन बच्चों, अमीनो एसिड। क्या बोल रहा हूं मैं? दिस इज व्हाट? दिस इज एन अमीनो एसिड। नाउ व्हाट सर? नाउ लेट्स मेक अनदर अमीनो एसिड। ओके, NH2CHRCOOH। ना अब इन दोनों की क्या करानी है? शादी, गठबंधन कराना। क्या कराना है? वी हैव टू डू दी गठबंधन। आई डोंट नो व्हाट वी से इन इंग्लिश, बट गठबंधन कराना है। तो गठबंधन कराना है तो दोनों कोई को क्या करना पड़ता है? दोनों कोई को कॉम्प्रोमाइज करना पड़ता है। यस और नो? बोथ द ब्राइड एंड द पर्सन हैज टू डू सम कॉम्प्रोमाइज। बेचारे लड़के का रूम छिना जाता है, लड़कियों की लाइक कुछ भी होता होगा। सो क्या करना? गठबंधन करना। तो जब तुम गठबंधन करोगे तो यहां से क्या करोगे? H2 निकालोगे। यहां से क्या करोगे बच्चों? यहां से H2 निकालोगे, H निकालोगे, सॉरी, यहां से क्या निकालोगे? H2 निकालोगे। और इधर से तुम क्या करोगे? इधर से तुम OH निकाल लोगे। इधर से तुम क्या करोगे? OH निकाल लोगे। तो बेसिकली व्हाट वी हैव डन इज दैट वी हैव रिमूव्ड वन मॉलिक्यूल ऑफ वाटर। मैंने कुछ गलत किया क्या? वी हैव रिमूव्ड वन मॉलिक्यूल ऑफ वाटर। H3O हो गया ये तो बच्चों, H3O हो गया। यहां से एक H निकलेगा। ओके, यहां से एक H निकलेगा। रुक जाओ भाई, रुक जाओ, बता रहा हूं, एक सेकेंड। चलो देखो, सो एक H ऊपर बता दूं, एक H यहां बना देता हूं। ये वाला H को तुमने भगा दिया और ये वाला H यहीं पे रहेगा। ठीक है? सो पेप्टाइड बॉन्ड कैसे बन गया? How you have made the peptide bond? By removing the water molecules. Yes or no? By removing the water molecules, you have made the peptide bond. Now see here. Now see here. अब इसको हम हटाते हैं। ओके, इसको जरा सा let us remove the things. तो यहां से क्या हटा? ये OH हटा दूं और यहां से ये हाइड्रोजन हटा दूं और इन दोनों को क्या कर दो? जोड़ दो। इन दोनों को व्हाट यू डू इज दैट, कुछ हो गया? कुछ तो हो गया बच्चों। अब ठीक है क्या? वन सेकेंड। ये मेरी क्लासेस में ना हमेशा ऐसी ऐसी हरकतें होते रहती है, बताओ। वन सेकेंड। चलो मैं स्क्रीन को थोड़ा सा बड़ा कर इधर खिसा घसका दे रहा हूँ। ठीक है, जस्ट वन सेकेंड। देख रहे हो ये क्या क्या हरकतें होती है मेरी क्लास में? तो वन सेकेंड। चलो, नाउ इट्स फाइन। ओके, नाउ इट्स फाइन। सो इनका शादी करवा देता हूँ, गठबंधन हो गया। अब ये दोनों खुशी खुशी मिलके रहने लगे। So this is what? This is a chain of protein. Like not only 2, 3 or 4. Shivesh, why you are crying? Not only 2, 3 or 4 will be there. Okay, 4, 5, 6, 7, 10, 11, 12, whatever. बहुत सारी शादियां करते हैं ये लोग, बहुत सारे। Okay, done. Now see guys, now see. This end is known as what? This end is free. This end is free. It is not having any bond. So this end is known as what? This end is known as N-terminal. This end is known as N-terminal. And in the end, this terminal will be free. COOH will be free. So this will be known as C-terminal. This will be known as C-terminal. Yes or no? Yes or no? Clear? So this side N-terminal and this side C-terminal. Clear to everyone? Clear to everyone? Any doubts anywhere? Let me know in the comment section. Great. Now let's see here. Let's talk about the types of amino acids. You know, in 2020 इन 2020 बच्चों ये अमीनो एसिड्स का स्पेलिंग AOOM होता है या सिंगल M होता है, बताओ तो जरा मुझे, अदरवाइज तुम मेरा मजाक उड़ा दोगे। अरे सर देखो सर, आपको अमीनो एसिड का स्पेलिंग नहीं आ रहा सर, पीटूंगा मैं ऐसे मजाक उड़ाओगे तो, समझ गए ना। चलो let's talk about the amino acids. How many type of amino acids? Sir, neutral, acidic and basic. Okay, how many types? Neutral, acidic and basic. So let's talk about the neutral amino acids first. Okay, neutral means what? Neutral means what? Equal amount of bases and acids are present. Okay, let's talk about the neutral amino acids. So example is what? Example is valine. Example is valine. Next is what? Next is alanine, and next is what? Glycine. Okay, इनका जरा structure याद कर ले. Alanine and glycine, the structure has been given in the NCERT. So let's read it out. Let's understand it. Okay. So glycine is what? Glycine is simple amino acid. Okay. Glycine is simple, just like our grandparents. They don't need anything. They just need what? They just need our love. Okay. So glycine is very simple, just like our grandparents. So how it can be simple, sir? To be simple, these groups to will be present at any cost. Yes or no? ये तो होगा ही होगा. Yes or no? बताओ. अगर ये नहीं होगा तो अमीनो एसिड्स कैसे बोलोगे? अगर एक इंसान को इंसान कहना है तो ऐसे दिखना पड़ेगा ना. अब वो जानवर जैसा दिख रहा है, तुम इंसान तो बोल नहीं सकते हो, तो ये तो रहेगा ही. बट सिंपल में व्हाट? सिंपल में बस एक छोटा सा हाइड्रोजन दे दो, ग्लाइसिन खुश हो जाएगा. एक छोटा सा हाइड्रोजन दे दो, ग्लाइसिन खुश हो जाएगा. So this is how you can learn the structure. Hi Krishma, how are you? This is how you can learn this structure. Yes or no? This is how you can learn the glycine का structure. Now what sir? Alanine का याद करवा दो. Alanine is what? Alanine is our grandmother. She is simple but she needs some jewelries. Yes or no? She is simple but she needs some jewelries. So थोड़ा सा इनको क्या करो, ऐसे ज्वेलरी दे दो. ठीक है, भले ही नकली दे दो तुम, लेकिन ठीक है दे दो. अब नकली दोगे तो फिर तुम्हें पिटाई पड़ेगी, उसकी रिस्पॉन्सिबिलिटी मेरी नहीं है. राइट. सो लेट्स सपोज दैट यू आर मेकिंग अ एलेनिन थोड़ा सा लिटिल बिट complex. So how much complex? Very little bit, sir. So let's add methyl group. Let's add methyl group here. Okay, this is what? This is aniline. This is alanine. ALANINE. Okay, this is alanine. Hi Anjali, how are you? I like that Sinchan DP. Chalo, this is alanine. Clear? Yes. Okay. Now what, sir? Now is serine. One another example of amino acid is let's suppose that, let's talk about the serine also. Serine is not a basic amino acid, neither it's an acidic, but serine is what? Serine is an amino acid which has alcohol in it. Serine, when you drink the alcohol, you are having very peace, right? So serenity, beautiful, peace, serine. So it has what? It has alcohol. It has alcohol in it. Sanjeev, very good morning. Sorry, good evening, bachcho. Good evening. It's good evening, okay? So we have this one, okay? Right? This is what? This is serine. Clear? Is it fine to everyone? Is it fine to everyone? Whoever is watching this lecture, hit the subscribe button. Hit the subscribe button, bachcho. Now see, let's talk about some of the basic amino acids. Let's talk about the basic amino acids. You know what? What NEET has done? NEET has given the same question in biology also and in chemistry also. And NEET वाले को बोले, अरे इनको क्या पता चलेगा? इनको क्या पता चलेगा कि एक ही क्वेश्चन को मैंने दो सब्जेक्ट में दे दिया. So can you see this? This is the level of repetition NEET is doing. So maybe in your case, some of the PYQs can be repeated. So भाई practice क्यों नहीं करते? PYQs किया करो. Basic amino acid is what? Basic amino acid is lysine and arginine. Basic amino acid is what? Lysine and arginine. Basic amino acid is lysine and arginine. Clear? NINE. Clear? Okay. Now don't depend for the spelling on me because I can write spelling very wrong. Okay, clear? Lysine and arginine, basic amino acid, and then we have as neutral amino acid as valine, alanine and glycine. Alcoholic amino acid as serine. Clear? Done, everyone? Chalo. Next is what? Acidic amino acid. See, if you're gonna ask me any trick for this acidic amino acid, I literally am gonna kill you. Why? Because नाम में ही बच्चे acidic glutamic acid और क्या aspartic acid, इसमें भी ट्रिक बताऊं अब तुम्हें? हाँ, तो बताता हूँ, बता दूं बच्चों, अगर नाम के आगे एसिड हो ना तो वो क्या हो जाएगा? अमीनो एसिडिक अमीनो एसिड. ठीक है, खुश हो गए? चलो, लेट्स मूव ऑन. Let's move on. So this is what acidic amino acid. Acidic amino acid. I'll write again. Acidic amino acid, right? So the example for this is what? Glutamic acid. Glutamic acid. अच्छा, ये दिख नहीं रहा होगा तुम लोगों को ना, मैं थोड़ा सा इधर हो जाता हूँ. And what? Aspartic acid. Aspartic acid. These things तो बच्चों you have to remember it at any cost. You have to remember because they all are given in the NCERT. Clear? They all are given in the NCERT, so you have to remember. Okay, chalo, great. Any doubts anywhere बच्चों? Any doubts anywhere till now? Any doubts anywhere? No? Okay. Now let's move on and let's talk about the another type of amino acid, which is aromatic amino acid. Which is aromatic amino acid. Yes, glutamic acid. Very good, Anjali. Very good. Aromatic amino acid. Aromatic is what? है ना? Beautiful smell. So aromatic amino acids are what? Those type of acids which has aromatic group. Example is phenylalanine. फिनाइल से स्मेल आता ना, ऐसे याद कर लो. फिनाइल से क्या आता? फिनाइल से स्मेल आता. सो फिनाइल एनालीन, फिनाइल alanine, phenyl alanine, right? Yes or no? And next is what? Tryptophan. Next is what? Tryptophan. Okay, tryptophan. फिनाइल से क्या होता? स्मेल आता, फिनाइल एलानिन, और उसके साथ किसको लगा दो? ट्रिप्टोफैन को लगा दो. क्लियर? ओके. कैसे? फिनाइल से स्मेल आया, इसको भी याद करवा देता हूं. देखो, फिनाइल से स्मेल आया, स्मेल बहुत गंदी थी, तुमने क्या किया? फैन ऑन कर दिया. फिनाइल से स्मेल आया, स्मेल बहुत गंदी थी, तुमने फैन ऑन कर दिया. सो व्हाट इज दैट? फिनाइल एलानिन एंड ट्रिप्टोफैन. क्लियर? ओके, यस और नो? चलो. ओके, ग्रेट. one more we have. one more we have. Tyrosine. We have one more. Tyrosine. एक और है बच्चों. Tyrosine. Tyrosine. Okay. Tyrosine. Clear? Any doubts? Any doubts? Any doubts? No doubts? Okay, chalo. Now again we have sulfur containing amino acids. Sulfur containing. याद हो रहा है क्या तुम लोगों को? Are you able to learn it things या बस bouncing it? हाँ? Sulfur containing amino acids. One is methionine, and the other one is what? The other one is cysteine. Methionine and cysteine. Yes or no? Methionine and cysteine. Clear? Chalo. This much is clear about the amino acids. Right? Great. Chalo. Next what, sir? Next I am going to talk about some of the things about amino acids. See, we are going to talk about the Zwitter ion. Twitter ion nahi bachcho, Zwitter ion. Okay. What is it? Zwitter ion. Okay. One second. Yes. What is it? Zwitter ion. So what is Zwitter ion? Zwitter ion are those type of ions in which positive and negative charge or positive ion or negative ion or cation or anion is present in the same molecule. So see here, if I am going to talk about this C, and then here we have what? Here we have COOH, we have COOH, and then here we have what? NH3, yes or no? And then we have R, okay, and then we have H. So if suppose somehow we are going to remove the H from here, okay? Somehow we are gonna remove this H here. This eraser tool is not working for me. बताओ मुझे लैपटॉप से erase करना पड़ रहा है. So somehow we have removed this H. So this will be what? This will be negative and this will be H positive. Now this H positive will be like, मैं कहाँ जाऊँ? मुझे घर से निकाल दिया गया है. अब मैं क्या हूँ? एक अबला H positive हूँ. कहाँ जाऊँ? So इस बेचारे को देख रहे हो मैंने यहाँ गलत लिखा, तुम बताते भी नहीं हो। यहाँ NH2 लिखूंगा ना पहले, तो बच्चों NH3 क्यों लिखूंगा, कहाँ से बॉन्ड बनाएगा ये? देख ली मेरी केमिस्ट्री कितनी अच्छी है। चलो इधर आओ। So this H positive is what? This H positive is Abla H positive. He is crying. Where we have to go now? What I'll do? I'll die alone. So what? This NH2 is very emotional person. He said that, Aja. Now what happened? This made NH3 positive. So can you see this? Can you see this? NH3 positive and COO negative are present in the same one. Are present in the same one, right? So this is what? This is known as Zwitter ion. This is known as Zwitter ion. Okay, clear? Right? When it is formed when we add amino acid in a neutral medium. When it is formed, when it is added in a neutral medium. Clear to everyone? Neutral medium, right? Now, this amino acid is what? This amino acid is also called as amphoteric nature. Amino acid is having what? Amino acid is having amphoteric nature. It is having amphoteric nature, right? Acha bachcho, tomorrow we are going to have what? We are going to complete the human circulatory system. Yesterday we did breathing and exchange, and tomorrow we are gonna do human circulatory system. Okay, everyone? Okay. Let me know in the comment section, bachcho. Chalo, great. Great. So amino acid is what? Amino acid shows amphoteric nature. That means if you are going to add it in acid, if you are going to add it in acidic medium, this amino acid will behave as what? This will behave as base. This will behave as base. And if you are going to, okay Shivesh, if you are going to add it in basic medium, then it is going to behave as what? It is going to behave as acid. Clear? Clear? Basic medium, acidic medium में डालोगे तो कैसे behave करेगा? It will behave as basic. Clear? Any doubts anywhere? Ecology, okay Bhavik, I'll do the ecology also. Don't worry. Ecology also I'll start. Okay? Chalo, great. This much is clear? Now, there are some amino acids which are essential. Amino acids can be classified into two types. What two types, sir? One is the essential amino acid. And the other one is non-essential. Why non-essential? इसकी कोई इज्जत नहीं है क्या बच्चों? है भाई, इज्जत है. इतनी इज्जत है कि ये खुद ही बॉडी बना लेता है. It is so important for our body that it makes खुद से. So if the body is making this amino acid by itself, so we don't need to eat it from outside. So we don't need to depend on diet. Okay? See what I am writing. Diet का spelling देख रहे हो मेरी? हाँ? 1 second बच्चों, तुम तो मजाक उड़ा दोगे मेरा. Chalo, I'll write here. Okay, we don't need to take it from diet, but essential amino acids are what? It is not made in our body. Body does not make it. Okay, but if I talk about the non-essential, body makes it, right? So if the body is making it, you don't have to eat, but if the body is not making it, you have to eat. Okay, so let's see the names of the essential amino acids. How you're gonna learn it? सारे याद कर लोगे क्या तुरंत में? बताओ मुझे. Will you be able to learn everything in just few seconds? छोड़ दो, ट्रिक ना बताऊं क्या? अरे सर बता दो. So I'll see. You went to a VIP lounge, okay? ऐसे ट्रेन में जाते हो तो रेस्ट रूम रहता है ना, जहाँ सामान वामान रख के करते हो. तो there was a person who was traveling without ticket. Okay, there was a person who was traveling without ticket, and he entered a VIP lounge, or I should say VIP hall. Where it entered the VIP hall. Okay, chalo. Now what, sir? Now he is shocked. दैट अरे वीआईपी हॉल में टीटी कहां से आ गया, वीआईपी हॉल में टीटी कहां से आ गया. व्हाई ही वाज शॉक्ड? बिकॉज ही वाज नॉट हैविंग एनी टिकट. सो वीआईपी हॉल में टीटी, वीआईपी हॉल में टीटी. सो दीज आर व्हाट? दीज आर एसेंशियल अमीनो एसिड्स. दीज आर एसेंशियल अमीनो एसिड्स. भाइयों यार ये तो याद कर लेना कि किसका ट्रिक है. पता चला इन एग्जाम यू आर कंफ्यूज लाइक ये एसेंशियल का था या नॉन एसेंशियल का था, राइट? चलो, सो वी फॉर व्हाट? वी फॉर वेलाइन. वी फॉर वेलाइन. शुड आई राइट इट कि तुम लोग लिख लोगे? चलो लिख देता हूं. वी फॉर वेलाइन. ओके, वी फॉर वेलाइन. आई फॉर आइसोल्यूसिन. I for isoleucine. P for phenylalanine. H for histidine. A for arginine. Arginine. L for leucine and L for lysine. Leucine, don't laugh बच्चों, ऐसे मेरे ट्रिक्स थोड़े अतरंगी होते हैं बट ठीक है चलो. L for leucine and L for lysine. M for methionine. Methionine. T for, मैं ही भूल गया क्या? टाई ट्रिप. हाय अंजलि, हाउ आर यू? अच्छा तुम तो पहले ही थी, पहले भी तो तुम आई थी. बताओ, मैं ही भूल गया. बताओ. टी फॉर टाई ट्रिप्टोफेन. ट्रिप्टोफेन. ओके, एंड देन दिस टी फॉर वेलाइन. अरे रुक जाओ एक मिनट बच्चों, ये क्या हुआ? वन सेकेंड, मुझे याद करने दोगे क्या? क्या था ये? ट्रिप्टोफेन था. एक और टी से क्या था? थ्रियोनिन. थ्रियोनिन, देखो बच गया मैं. थ्रियोनिन. चलो ठीक है. सो वी हैव वेलाइन, आइसोल्यूसिन, फिनाइल एंड दिस दैट. याद हो जाएगा एवरीवन? याद हो जाएगा? मजाक मत उड़ाओ मेरा, मजाक मत उड़ाओ मेरा, भूल जाता हूँ कभी कभी. चलो आगे बढ़ो. शुड आई मूव फॉरवर्ड? चलो अब आते हैं क्या? अब आते हैं नॉन एसेंशियल अमीनो एसिड्स को याद कर लेते हैं. नाउ लेट्स लर्न टायरोसिन. भाविक पहले नहीं बता सकते थे तुम? थैंक्स अंजलि, थैंक यू सो मच. अंजलि अब इस वाला ट्रिक के बाद तुम मुझसे गुस्सा हो जाओगी तो उसके लिए मैं पहले ही माफी मांग रहा हूं तुमसे. ओके ओके चलो. सो let's suppose that you have a girlfriend. Okay, you all guys have a girlfriend. Suppose, okay, you don't have, I know. Let's suppose that you all have a girlfriend. Now what? Now to impress your girlfriend, what you do is that you either take her to the fuchka date, golgappa, yes or no? Golgappa date you take it, or what you do, or you may take her to samosa date. Okay, samosa date. So samosa is what? Samosa date we will take. We will take samosa date. And what will happen whenever girls eat samosa, they got crazy. Whenever girls eat samosa, they got crazy. Okay. Sorry to all the girls, but क्या करूं बच्चों, ट्रिक्स बनाना होता ना. चलो 1 second. So I'll write here almost all girls go crazy after getting tasty potato samosa. Okay, almost all the girls go crazy after getting potato samosa. Clear? Okay, yes or no? बताओ मुझे. याद कर लोगे क्या? ठीक है. सो ए से क्या हो जाएगा? एक ए से हो जाएगा एलेनिन. 3 ए है यहाँ पे. ओके, 3 एज आर देयर. सो 1 एक ए से हम लिख लेते हैं एलेनिन. सेकेंड ए से हम लिख लेते हैं एस्पार्जीन. इसको दूसरे कलर से लिख देता हूं. ओके, एस्पार्जीन. फिर हमारे पास क्या क्या है? 1 सेकेंड बच्चों, लिख लोगे क्या ये कि मैं लिखवा दूं? पूरा लिखवाऊं क्या? बताओ, पूरा लिखवाऊं या लिख लोगे तुम? नॉन एसेंशियल एमिनो एसिड का लिस्ट लिख लोगे क्या? जी से क्या हो जाएगा? ग्लूटामिक एसिड. दूसरा जी से क्या हो जाएगा? ग्लाइसिन. ओके, ग्लाइसिन. फिर सी से क्या हो जाएगा बताओ? सिस्टीन. सिस्टीन हो जाएगा. ओके, यस और नो? फिर एक और ए है हमारे पास, तो एलेनिन हो गया, एस्पार्जीन हो गया, और क्या आ जाएगा, एस्पार्टिक एसिड, एस्पार्टिक एसिड। ओके, यस और नो, राइट, क्लियर। टी से क्या हो जाएगा हमारे पास, टायरोसिन, टी से क्या हो जाएगा हमारे पास, टायरोसिन। पी से क्या हो जाएगा बताओ बच्चों, प्रोलीन, प्रोलीन। एंड एस से क्या हो जाएगा, सेरीन, एस से क्या हो जाएगा, सेरीन। क्लियर, ओके। कितने हो गए, 1 2 3 4 5 6 7 8 9, अरे एक छूट गया, एक तो छूट गया बच्चों, क्या छूटा बताओ, ट्रिप्टोफेन, ट्रिप्टोफेन छूटा क्या, नहीं नहीं, क्या छूट गया, अरे बताओ बच्चों क्या छूट गया, चलो लेट मी चेक, लेट मी चेक, एक क्या छूट गया, लिखवा दीजिए सर। अच्छा, लिखवा दिया बच्चों, लिखवा दिया बच्चों, बताओ, तुम्हारी खुशी के लिए मैं लिखवा दिया, एक छूट गया बच्चों। ग्लाइसिन लिखा क्या मैंने, ग्लाइसिन लिखा क्या, यस आई हैव रिटन ग्लाइसिन। वन सेकेंड बच्चों, 1 सेकेंड, 16, एक छूट गया बच्चों, कुछ तो छूट गया, कोई बात नहीं, चलो मैं बता दूंगा तुम्हें कमेंट सेक्शन में क्या छूट गया, ठीक है। अभी के लिए मैनेज कर लो, अभी के लिए थोड़ा सा मैनेज कर लो, कर लोगे, कैसे नाराज तो नहीं हो जाओगे, सर आपने एक लिखवाया नहीं सर, अब क्या होगा हमारा जिंदगी, बताओ। जी से एक छूट गया बच्चों, मैं कमेंट सेक्शन में बता दूंगा, ठीक है। चलो ऐसे नाराज ना होते, ठीक है, आगे बढ़ो, आगे बढ़ जाते हैं हम लोग, ठीक है। मैं ही भूल गया, बताओ, ट्रिक याद हो गई मुझे, मैं ही भूल गया जी से। सेशन के एंड तक ना पता करते करते मुझे याद आ जाएगा, मुझे याद आ जाएगा तो मैं बता दूंगा तुम्हें, ठीक है। चलो ना, आ जाओ, आ जाओ, प्रोटीन। नेक्स्ट लेट्स टॉक अबाउट दी प्रोटीन स्ट्रक्चर्स, प्रोटीन स्ट्रक्चर्स, ओके। प्रोटीन स्ट्रक्चर्स के बारे में हम बातें थोड़ा कर ले। How much types of structures of protein are there? How much types of structures of protein are there? Sir, 3 types: primary, secondary and tertiary. Primary, secondary and tertiary. Yes or no? Primary, secondary and tertiary. यस शिवेश, कुछ नहीं कर सकता तो कमेंट में ही बताऊंगा मैं तुम्हें, अगर याद आ गया सेशन के बीच में तो मैं बता देता हूँ, ठीक है। बट लेट्स नॉट वेस्ट द टाइम, पता चला कि ये वन शॉट जो है 1 डे का सेशन हो गया, राइट। सो 1 डिग्री, 2 डिग्री और 3 डिग्री, प्राइमरी सेकेंड, तुम तो मजे ले लोगे 1 डे का, मैं बताओ मैं ऐसे खड़े खड़े एक दिन कैसे पढ़ाऊंगा, राइट। सो लेट्स मूव ऑन। 1 डिग्री, 2 डिग्री एंड 3 डिग्री, ग्लूटामिक एसिड आई हैव रिटन, शिवेश भाविक लिख दिया हूं मैं, कोई बात नहीं, अरे आ जाओ, अब आ जाओ, आ जाओ, आ जाओ इधर बच्चों, आ जाओ। 1 degree, 2 degree and 3 degree. So primary structure is what? Primary structure is just a linear structure. Primary structure is what? Just a linear structure. You have taken the amino acids, you have taken the amino acid and made a chain. That is known as what? That is known as primary structure. Okay. If I talk about the secondary structure, so secondary structure is of 2 types. Secondary structure is of 2 types. One is what? One is alpha helix. One is the alpha helix and the other one is beta plated. Alpha helix and beta plated. Now see here. Alpha helix is what? Just like your DNA. Okay. Alpha helix is what? Just like your DNA. Like this. And here, what is happening? Between these things, hydrogen bond is present for maintaining the stability. Yes. But beta plated is what? Beta plated. Hi Shrishai, how are you? You have your biology exam tomorrow. Great. Shrishai, where were you? हाँ, इतने दिन से कहाँ थे Shrishai? Now see, what about beta plated sir? So beta plated is what? Beta plated is just like we fold the t-shirts na, that's how it is folded. Let's suppose, do I have something? 1 second bachcho, I'm just coming, I'll bring something for you. Okay? 1 second, 1 second. 1 second bachcho. 1 second. चलो चलो चलो, आई एम बैक, आई एम बैक। कहाँ गया मेरा माइक, कौन किसने चोरी कर लिया भाई? ओके, जस्ट अ सेकंड, 1 second, माइक लगा लो। चलो, ग्रेट। सो सी हियर, बीटा प्लेटेड इज व्हाट? लेट्स सपोज दिस इज अ sheet, okay, व्हाट वी हैव डन, ये पूरा लीनियर, मान लो ये क्या है हमारे पास, लीनियर मॉलिक्यूल ऑफ अमीनो एसिड्स। ओके, अब इसको क्या किया मैंने, ऐसे प्लेट्स बना दिए, ऐसे प्लेट्स बना दिया, ऐसे ऐसे फोल्ड कर दिया, तो ये देख रहे हो यहाँ पे क्या बन गया, यहाँ दिस स्ट्रक्चर इज नोन एज बीटा प्लेटेड, और ये वाला लेयर और ये वाले लेयर के बीच में हाइड्रोजन बॉन्ड आ गया। इस तरह के स्ट्रक्चर्स को क्या बोलते हैं, बीटा प्लेटेड स्ट्रक्चर। बच्चों क्लियर है क्या? इज इट क्लियर टू एवरीवन? इज इट क्लियर? यस, बता दो मुझे कमेंट में। चलो, ग्रेट। शिवेश is saying hi. Sai, I am angry with you. Why Shivesh? Did Sai something steal something from you? चलो चलो, ग्रेट। नाउ अब इसको मैं बनाऊं कैसे, बीटा प्लेटेड को समझ जाओ बच्चों, तुम समझदार हो, ऐसे करके, राइट, like this, and this is what they are joined with the hydrogen bond. Okay, clear? Where it is present, beta plated molecules, where it is present, silk protein, okay, silk protein. मैं देख रहा हूँ मैं स्पेलिंग कितना मिस्टेक कर रहा हूँ, silk protein which is known as fibrin protein, which is fibrin protein. Okay, fibrin protein, and alpha helix is present where, ये जो तुम्हारे प्यारे प्यारे से बाल है ना, जिसको तुम जेल लगा के ठीक करते हो, वहां पे प्रेजेंट होता है अल्फा हेलिक्स। केरेटिन, किसमें, केरेटिन प्रोटीन, यस और नो, केरेटिन प्रोटीन। ओके ओके, क्लियर, ग्रेट ग्रेट। चलो, नाउ लेट्स मूव ऑन विद द टर्शियरी स्ट्रक्चर, लेट्स मूव ऑन विद द टर्शियरी स्ट्रक्चर। व्हाट इज टर्शियरी स्ट्रक्चर बच्चों? सी, if suppose there is alpha helix and alpha helix कुछ ऐसा था, let us understand this example with the help of alpha helix. Okay, so alpha helix, whoever is watching this video, subscribe to this channel. Okay, चलो, so alpha helix was like this, ठीक है। अब इस alpha helix को what I have done, if इस alpha helix को और मैंने क्या किया, coiling कर दी, alpha helix को और coiling कर दी मैंने, then it will make what, it will make tertiary structure, right, it will make tertiary structure। ये कुछ बहुत ही गंदा सा बन गया, बट ठीक है, तुम समझ जाओगे। and in this tertiary structure, to maintain what, in this tertiary structure, what we have to do is that we are going to have electrostatic bond, we are going to have electrostatic bond. Okay, remember in these cases guys, in these cases hydrogen bond was the one who was stabilizing the things, right? But here what is happening, here electrostatic bond also came to stabilize the molecule, and hydrogen bond will be to present, hydrogen bond to in helix ke beech mein it will be present। clear to everyone? yes or no? yes or no? let me know in the comment section guys, let me know in the comment section। clear? great great, very good very good very good। okay, now where these are present sir, tertiary structures are present in enzyme, hormones, and what sir, just enzyme and hormones। so all the enzymes, all the hormones in our body is showing what, is showing the tertiary structure। clear? great great bachcho, any doubts anywhere? Shivesh, any doubts? Sri Shai, any doubts? Anjali, Bhavik, any doubts? Any doubts bachcho? No doubts? great, chalo, now we have another one which is quaternary structure, another one which is quaternary structure। what is this quaternary structure? see here, if I am going to talk about the quaternary structure, so it is more coiling of tertiary structure, right, more coiling of tertiary structure, and the only example of quaternary structure which has been given in our NCERT is what, it is hemoglobin, it is hemoglobin। okay, so it is hemoglobin। clear? this is what, this is the quaternary structure। okay, and lots of bonds are there, you don't have to worry about the structure of this thing, you just have to remember example of this is what, hemoglobin। clear? any doubts anywhere? Great, great. So in this, you also remember electrostatic bond is also there. Okay, electrostatic bond is also there. And one more, वो केमिस्ट्री में तुम बहुत ही भयानक सा नाम लेते हो ना, क्या नाम है उसका, वंडरवॉल बॉन्ड. वंडरवॉल फोर्सेस सर, नॉट बॉन्ड, वंडरवॉल फोर्सेस. वंडरवॉल फोर्सेस, वीक फोर्सेस, वो भी यहाँ पे होता है. हाइड्रोजन बॉन्ड भी यहाँ पे होता है. सो इफ इन केस क्वेश्चन कम्स दैट व्हाट बॉन्ड्स आर stabilizing the structure of quaternary structure, so here are the list of the bonds. Clear? Okay, yes or no? 2 alpha 2 beta, yes yes yes, 2 alpha 2 beta units, 2 alpha and 2 beta units से बना होता है. hemoglobin is made up of 2 alpha and 2 beta units, and बीच में क्या होता है, iron. Yes or no? Just looking like a wow. Thank you so much Shivam. Thank you so much Shivam. Chalo, clear? Okay, now let's move on. Let's move on, and this I think so we have completed biomolecules. Nahi sir, we have just completed the protein part bachcho. Now let's talk about the carbohydrates. Now let's talk about the carbohydrates. Ready? You want to take a break? Let's take a break to drink water. Okay, let us drink water. Let us drink water. वरना तुम बोलोगे सर पानी भी पीने नहीं देते. Okay, now let's move on. Let's move on. Red color देता है. Yes, yes, exactly. Red color, oxyhemoglobin, all those things. Shrisha, we discussed yesterday, breathing in exchange. Okay, chalo. Now let's talk about the carbohydrates. Carbohydrates. Now what is carbohydrates, sir? Carbohydrates are known as sugar. Carbohydrates are known as what? They are known as sugar, right? Yes. And if I talk about the carbohydrates, so carbohydrates are basically what? Made up of carbon. Yes, obviously it is an organic, it is a biomolecule, so it will be made up of carbon. Yes or no? 5-second break. Yes, exactly. 5. Yes, yes, Shivam. So it is a carbohydrate. We don't need a break, guys. Don't need a break, right? 3 घंटा तो ऐसे ही तुम्हें बैठने की आदत होनी चाहिए. चलो पढ़ो, आ जाओ, आ जाओ. So carbohydrate is what? Carbohydrate is a compound which is made up of carbon. So it may be made up of 2 carbon also. Yes or no? It may be made up of 3 carbon also. It may be made up of 4 carbon also. But sir, whenever we see whenever we classify the carbohydrate, we write monosaccharide. Yes or no? We write monosaccharide. We write disaccharide. Yes, and we write polysaccharide. And when we talk about the number of carbons, sir, so in monosaccharide we are writing 3 to 7. We are writing 3 to 7. Why not 2? Why not 2, bachcho? Why we are not saying that 2 carbon molecules can also make carbohydrates? So when you are going to call 2 molecules, these 2 molecules will not show the property of carbohydrates, will not show the property of monosaccharide. That's why the limit for carbohydrate is 3 carbon. Okay, yes or no? Yes or no? And if you need more clarity, I'll ask Nikhil sir to make you understand why it's not showing the properties and all. Okay, right? Chalo. So now let's read the monosaccharide first. Let's read the monosaccharide first. Okay, monosaccharide first. How many of you are has this thing that आज तो इस chapter को खत्म करके ही जाना? How many of you? How many of you? जैसे मैंने बोला एक बच्चा भाग गया, सर हमसे तो नहीं हो पाएगा, right? चलो चलो, next let's be here and let's try to finish off this chapter, because this chapter is going to give you lots and lots of marks. Okay, lots and lots of marks. Monosaccharide, if 3 carbon is gonna there, then bachcho we are going to call it as triose sugar. Then we are going to call it as triose sugar. And if we are going to talk about 4 carbon, then tetrose, and 5 pentose. So we have to remember, we have to learn the pentose. And don't worry, all the things, all the structures today I'm gonna make it learn. Okay, you don't have to worry. 6 carbon is hexose. 6 carbon is hexose and 7 carbon is what? Heptose. Yes or no? Okay. Now, this was what? This was the monosaccharide. Now, if I talk about the disaccharide, so disaccharide is what, sir? Disaccharide is what? It is made up of 2 monosaccharide. Disaccharide is made up of 2 monosaccharide. So, for example, monosaccharide example is glucose. Example is what? Glucose. One more example, anyone? Anyone? Any other example? Sir, fructose. Fructose. More examples, sir. Galactose. Yes, galactose. शिवम तुमने मुझे स्पेलिंग बुलवा दी. Galactose, glucose, fructose, galactose. Yes or no? Now see here. These are what? These all are the hexose sugar. Any example of pentose sugar? Sir, ribose and deoxyribose. श्रीशा, glycosidic bond? हाँ वो तो पढ़ेंगे हम. Ribose and deoxyribose. Ribose and deoxyribose. Clear? Okay. Now what we are going to do is that we are going to take 2 molecules of monosaccharide. Let's take glucose and glucose. Okay, let's suppose we take glucose and glucose. So this glucose and glucose is gonna make what? It is gonna make disaccharide, right? Yes or no? And the glucose and glucose is basically what? It is maltose. When 2 glucose molecule is gonna meet, they will be known as maltose. Okay, they will be known as maltose. Clear? Is it lagging, bachcho? Is it lagging? No? See, video may lag for some time, but my voice will be clear to you, right? So you can manage. Thoda to manage kar lo. Now see here. If suppose we have what? We have glucose plus fructose. We have glucose plus fructose. Then it will be called as what? It will be called as sucrose. Acha. So Shrisha will be like, I will answer this first. Okay. Yes, chalo. I'll ask. Which bond is gonna present here, sir? The bond which is going to present here will be glycosidic bond. Glycosidic bond. Is it so? Is it so, bachcho? Will it be glycosidic bond? Will it be glycosidic bond? Yes. The bond will be what? Glycosidic bond. Clear? Chalo. Now we have glucose and fructose. This will be called as what? Sucrose. This will be called as sucrose. And then glucose and galactose. Glucose and galactose. It will be called as what? Lactose. Lactose. याद कैसे करना सर? याद कैसे करना सर? See, glucose and glucose. Malt, you know, malting process, sugar cane, glucose, sugar cane is very meetha. Glucose, sugar cane very meetha. Malting process, maltose, ऐसे याद कर लो. Now, glucose and fructose, sucrose. Fructose, sucrose, right? Then what? Galactose, lactose. So see, glucose is common in all of them. So what you have to do is that glucose, maltose, sucrose में क्या आ जाएगा? Fructose आ जाएगा. See, this part is similar ना? ये part थोड़ा similar है. Lactose में ये part similar है, right? Will it be? You will be able to learn it? Yes. Okay. Now, in this case, 1 second, I'll drink a sip of water. Okay, great. Now, in this case, what we are going to do is that next we are going to talk about the polysaccharide. Right? Don't worry, guys. Till 8 o'clock, we will be able to complete this chapter. Don't worry. Okay? We have this polysaccharide. So, if I talk about the sucrose is easy, right? Yes. We have this polysaccharide. So, polysaccharide is what? It is more than 10. बहुत सारे लोगों से इन्होंने शादी कर ली. More than 10. Okay? More than 10 monosaccharide makes the polysaccharide. Now see, this polysaccharide is what? It is for storage. And it is for structure. So see here, like I am little fat now. Okay, when I was a kid, I was very thin person. Now I am little fat. So what is happening? what is happening? I am storing lots and lots of glycogen. I am storing lots and lots of glycogen. Yes or no? Glycogen. So glycogen is an example of polysaccharide. Now, one more thing. There is a plant in my house that I usually ask that plant to do yoga and all of those things, go to gym, but that plant is not listening to me. So that plant is also storing and getting fat. What storing, sir? It is storing starch. It is storing starch. Yes or no? It is storing starch. Clear? Clear? Now, structural. These are what? These are the storage thing. They don't make anything. Glycogen is not making anything. It's just giving me fat, right? Starch is not making anything. It's just being stored in it. But structure is what? These are what? Making some structures like cellulose. Yes, Sri Shai. Very good, Sri Shai. Next is cellulose. Okay, cellulose makes what? Our cell wall. No, sir, we are not plants. Plant cell wall. Yes or no? Plant cell wall. Now, if I talk about the chitin, where is present chitin? Chitin कहाँ present होता है? Let me know in the comment section, बच्चों. Let me know about in the comment section where chitin is present. Chitin is present in the cell wall of fungi. Yes or no? Next we have peptidoglycan. Next we have peptidoglycan. So peptidoglycan, where it is present? It is present in the bacteria, membrane of bacteria. Yes or no? Clear? Chalo, let me know in the comment section if this thing is clear to everyone. If this thing is clear to everyone. If this thing is clear to everyone. बताओ मुझे. See, next is what? Next we classify on the basis of reducing sugar and non-reducing sugar. Okay, reducing sugar and insects, arthropods, yes. Reducing sugar and non-reducing sugar. See, in this thing, you don't have to learn how reducing, why reducing. You just have to know that this is reducing, this is non-reducing. So see here, what you have to do is that sucrose is non-reducing. Just learn this thing, okay? Just learn this thing. Sucrose is what? PE class बोले तो? PE class बोले तो? अच्छा, ऐप पे? Yes, yes, we have 9:30 we have. चलो, कहाँ गया मेरा सुक्रोज? तुम लोग ऐसे चोरी कर लिए क्या सुक्रोज को? हाँ, सुक्रोज को चोरी कर लिए क्या भाई? कहाँ चला गया? Yes, this sucrose is what? Sucrose is non-reducing. This sucrose is non-reducing. And monosaccharides are what? Monosaccharides are reducing. All the monosaccharides are reducing. So you just learn it. Monosaccharides are reducing. Sucrose is non-reducing. And polysaccharide is what? Polysaccharide is also non-reducing. Okay. Polysaccharides are also non-reducing. What about them, sir? What about this, which is lactose and maltose? So they are what? They are reducing. Lactose and maltose are reducing. Okay. Chalo, great. Great. Now let's move on. Now let's move on and let's talk about the structures. Okay. Let's talk about the structures of monosaccharide. Structures of monosaccharide. Okay. So in the structure, first we are going to talk about the ribose sugar and then deoxyribose sugar, and then we are going to talk about what? Glucose, and then disaccharide and polysaccharide. Then the carbohydrates will be over, and as soon as the carbohydrates is over, guys, our chapter is finished 80%, right? Our chapter will be finished 80%, then very less part will be left. Okay, chalo. So now let's talk about the monosaccharide. So what is monosaccharide? For example, if I talk about monosaccharide, the example which is ribose sugar, So see, I am going to help you to make the structure. So see, ribose sugar is what? 5 carbon structure, right? 5 carbon structure. So let's make the 5 carbon chain first. 1, 2, 3, 4, 5. How many carbons we have here? 5 carbons. Next what? Next we have, here we have CHO. Yes or no? Here we have OH, here we have OH, here we have OH, and here we have OH, right? This carbon will contain H and this carbon will contain H, and rest every carbon will have what? Hydrogen. Not the first carbon. First carbon mein to valency is complete. So let me remove that mark which I have made. Let me remove that mark which I have made. One second guys, 1 second. Chalo, we have removed this mark. So what is happening, sir? Here this ribose is this is the linear structure of ribose. But sir, we have to make the circular structure. So how we are going to make the circular structure here? See, the second last carbon is going to attack on this oxygen. The second last carbon is gonna attack on this oxygen, and what is going to happen? This kind of structure will be formed. See, this is the oxygen, and fourth carbon is attacking. So from this oxygen, fourth carbon will be joined, and on the fourth carbon we have the fifth carbon. So let's make the fifth carbon here, and let's talk about the CH2OH, right? CH2OH. And then here we have what? Here we have the third carbon, here we have the second carbon, and here we have the first carbon, okay? Right? Yes. So this is the basic structure. Now on the first carbon you write OH, on the second carbon you make OH, on the third carbon you make OH. Yes, this is the structure of the ribose. This is the structure of the ribose. Is it clear to everyone? Yes or no? Yes, bachcho, is this clear? Yes? Great. Great. If this is clear, then it's very good. If this is clear, bachcho, it's very, very good. Chalo. So, now sir, if we need to make the deoxyribose sugar, what we have to do is that just remove the O from here. If we are going to remove the O from here, if we are going to remove, not OH, bachcho, just the O, okay? Just the O. If we are going to remove just the O here, what we are going to get? We are going to get deoxyribose sugar. What we are going to get? Deoxyribose sugar. Clear? Is it clear? A very easy method. A very easy method. Now, let's see. Let us remember the structure of glucose also like this. Let us remember the structure of glucose. बता दूं क्या structure of glucose में? ऐसे याद हो जाएंगे तुम्हें? चलो बता देता हूँ. Just few more minutes. When we are gonna complete carbohydrates, when we are gonna complete, then almost half of the chapter will be over, right? Half, or I think 80% of the chapter will be over. Then तो छोटी-छोटी चीजें करनी है फिर. See, glucose. Let's talk about the glucose. Is my video lagging or is it fine, bachcho? Okay, 1 second. Just a second, guys. Just a second. Now I think so it's fine. Ab theek hai kya? Yes, now it's fine. Okay. 1 second. I'll check. Chalo. Now it is fine. Bachcho. Chalo. So let's get back to the glucose. Glucose is again made up of 6 carbon. Glucose is made up of 6 carbon. So 1, 2, 3, 4, 5, 1, 2, 3, 4, 5 and 6, right? So in the first carbon what we have? Again this group, CHO group we have, right? Second carbon what we have? OH. Third carbon is what? ऐसे बोलते ना, तीन तिगाड़ा काम बिगाड़ा. तो third carbon में OH is on different side. So this is the structure of a linear glucose. Again, what we have to do is that make the cyclic structure. So how we are going to make the cyclic structure? See, which carbon is going to attack? The second last carbon. This carbon is going to attack to this carbon, okay? When they're going to attack, they are going to have this structure. Let's make the O. So from O, which carbon will be attached? From O, the fifth carbon will be attached. And on the fifth carbon, we have what? we have the 6th carbon, so let's make the 6th carbon here, right? And now let's complete the structure. So 1, 2, 3, 4 and 5 carbon. Okay, now see, OH is present where? Always remember, OH will be present down, but on 3rd carbon it will be up. Clear to everyone? On 3rd carbon OH will be up, rest sub mein OH will be down. Yes or no? Let me know in the comment section if this thing is clear. Everyone. Clear? Great. Chalo. Now let's move on and let us talk about the disaccharide bonds. See, I have made monosaccharide. So 2 monosaccharides will make what? 2 monosaccharides will make disaccharides. Yes or no? So as Shri Sai said that disaccharide is containing what? Glycosidic bond. So let me make a very quick glucose molecule. So this is one glucose molecule. It is having OH here. This is having OH here. And this is having OH here. This is having CH2OH. Similarly, I'm going to make another glucose molecule. Okay, I'm going to make another glucose molecule like this. Hi Radhika, how are you? Another molecule I'm going to make. Okay. So this is what? This is the another glucose molecule. Here let's have OH. Yahan ka OH will be upward. This will be OH. And this will having CH2OH. Now sir, how the bond is created? So see here. Yeah, Radhika came early. Is that so? Bhavik, ऐसे नहीं बोलते. चलो इधर आओ. So now what we are going to do is that we are going to remove water. And when we are going to remove water, then glycosidic bond will be formed. Then what bond will be formed? Bachcho, then glycosidic bond will be formed. So let me remove this water here. One second. Yes. So let me remove this water here, and what we are going to get? We are going to get one oxygen. एक oxygen तो बच जाएगा ना? So see, this is what? This is the alpha glycosidic bond. Okay? Alpha glycosidic bond. Sir, what is alpha? What is beta? See here. If 1 second, मुझे बताना चाहिए था ये, but मैं अभी बता दे रहा हूँ. If this is what? If OH is down on 1st carbon, if OH is down, then alpha, and if OH is up in 1st carbon, then it is known as beta. So here what we have done, here we have taken 2 alpha hydrogen. So we are gonna say it as the bond is alpha glycosidic bond. And which carbon? This is 1st carbon and this is 4th carbon. So the bond is between 1st carbon and 4th carbon. So we are going to say 1,4 alpha glycosidic bond. Yes or no? Clear? Guys, I have full one-shot sessions for you. We are going to complete human physiology in the next week. Yes or no? शिवेश, then और मजा आएगा तुम्हें. तब और मजा आएगा तुम्हें. Yes? चलो, great. Clear to everyone? Now let's go back to the polysaccharide, and let me talk about some of the structure. ये glycogen is made up of what, sir? Glycogen is made up of glucose. Lots and lots of molecules of glucose makes the glycogen. Starch is made up of what? Starch is made up of 2 things, amylose and amylopectin. Starch is made up of what? Amylose and amylopectin. मैं यहाँ पे लिख रहा हूँ उसको, okay? I am writing starch here. 1 second. अच्छा, do you guys need notes for this? Notes चाहिए क्या इनका? Whatever I have written, do you want this? I'll add it. I'll add the link in the comment section. a Google Drive link so that you all can access the notes. Will that be fine? Chalo, great. So we have what? If I talk about the starch, so starch is made up of two structures. One is amylopectin, and the other one is amylose. Okay. So amylose is what? Goal amylose. Amylose is what? Goal amylose. Round amylose. Okay. Amylose is what? Goal amylose. Round amylose. Basically, alpha helix structure. Okay. Clear? Amylopectin is what? Branched. Amylopectin is what? Branched. Now see here, bachcho. If I'm going to talk about the starch test, so what happens there? Iodine gets stuck. So iodine gets stuck in which part? Amylose or amylopectin? Let me know in the comment section. Amylopectin may iodine is stuck or it is stuck in amylose? Is it stuck in amylopectin or is it stuck in amylose? Tell me in the comment section. It is stuck in what? It is stuck in amylose. Here the starch is stuck, and this gives the starch test. Okay, this gives the starch test. Clear? Sorry, iodine test. Clear? Okay, chalo. Now what, sir? Now see, on one more category, we are going to divide the polysaccharide. We are going to divide polysaccharide in one more category. Sir, the category is heteropolysaccharide and homopolysaccharide. Okay, homopolysaccharide. So homo तो we saw. होमो में क्या क्या आ जाएगा? ग्लाइकोजन आ गया, स्टार्च आ गया, सेल्यूलोज आ गया, ओके, एंड क्या आ गया यहाँ पे? काइटिन आ गया, राइट? काइटिन इज मेड अप ऑफ व्हाट? काइटिन इज मेड अप ऑफ NAG. काइटिन किससे बना? नाग से. NAG, N-acetyl glucosamine. NAG से काइटिन बना हुआ. Now heteropolymer is what? Those polysaccharide which is made up of two or more than two monomers, which is what? Which is peptidoglycan. Now where this peptidoglycan is present? Anyone in the comment section? शिवम भाविक, कहाँ प्रेजेंट है ये पेप्टिडोग्लाइकन? कहाँ प्रेजेंट है ये पेप्टिडोग्लाइकन? This peptidoglycan is present in the bacterial cell wall. And this is made up of what? Yes, Bhavik, very good. This is made up of what? NAG and NAM. Okay, this is made up of NAG and NAM. Clear to everyone? Yes or no? Yes? Chalo. One more thing we have to figure it out about inulin. Inulin is what? Once it has given in the option in one question. So inulin, many of us get confused with the insulin. So it is what? It is not insulin. It is a type of carbohydrate. Okay, it is a type of carbohydrate. Inulin is what? It is a type of carbohydrate. Just remember this much. Okay, just remember this much, that it is a type of carbohydrate. Clear? Great. Okay, now most abundant biomolecule on the earth. Guys. Most abundant biomolecule is cellulose. Most abundant biomolecule is cellulose. Okay, is cellulose. Now most abundant protein is what? Most abundant protein we have is rubisco. It's given in the NCERT. Okay, rubisco. Most abundant protein is rubisco. Most abundant animal protein is what? Most abundant animal protein is collagen. Okay, screenshot this thing, because this can be asked in the exam. Okay, so we are done with the protein and we are done with the carbohydrates. Now what we have? Now we have lipids. Should we do it? Kar lo pura? Should we complete it, guys? Let me know. 1 second, ये क्या हो गया? Okay. Should we complete the chapter? Yes, obviously. It's one shot, sir. We are gonna complete it. We are definitely gonna complete this one. Yes or no? Chalo. Chalo. Let's see here. Let's see here. Let's talk about the lipids. Let's talk about the lipids now. Sir, what is lipids? Lipids are those, is it biomacromolecule or is it, hi Sakshi, how are you? Is it biomacromolecule or is it biomicromolecule? So sir, lipids are what? Lipids are biomicromolecule, but they are acid insoluble. Why? Because they are making vesicles and they are not able to cross the cheesecloth. Yes or no? Yes or no, bachcho? Yes, chalo. So lipids are biomicromolecules. Biomicromolecules, but they are what? They are water insoluble. They are water insoluble. Yes or no? Yes, sir. They are water insoluble. Inulin is hetero. Inulin is hetero. Okay? Inulin is hetero. Chalo. Great. Now see here. Let's talk about the types of lipids. Let's talk about the types of lipids. One is simple lipid, okay? One is simple lipid. Simple lipid are of what? Simple lipid is what? It is fats and oil. In simple lipids, what do we have? We have fats and oils. In simple lipid, we have fats and oils, okay? Fats and oils. So see here, if I talk about the oils, so oils are what? Oils have low melting point. Oils have low melting point. And if I talk about the fats, fats have, yes, Sakshi, you were saying something? Fats have high melting point. Fats have high melting point, okay? Yes or no? Yes. Now, examples of oil which is given in the NCERT is gingerly oil. It is what? It is gingerly oil. Clear? Now, I don't know about the spelling. Gingerly oil. देख लेना अपना spelling, ठीक है? Gingerly oil. And if I talk about the fats, so mainly what? Mainly it is triglycerides. Mainly the fats we're going to study is the triglycerides, okay? Okay, yes or no? Clear बच्चों? Clear बच्चों? चलो. Oh, inulin. Polysaccharide. It's a polysaccharide, Sakshi. So, अरे क्या बोल रहा हूँ मैं? Sakshi. देख रहे हो मेरे को? हाँ? So, fats are what? Fats are basically triglyceride. Yeah, I got it. Triglyceride. So, inulin is basically what? See, Sakshi, यहाँ पे लिखा हूँ मैं inulin. Inulin basically what? It's not in the NCERT, but once what happened? Option was given. So, many of us can get confused with insulin. So, inulin is not an insulin, it is a carbohydrate. Just remember this, okay? Just remember this. बाकी बहुत ज़्यादा questions नहीं आएंगे इससे. Now, see here. Next what we have? Next we have conjugate lipids. Now, what is conjugate lipids? Conjugate lipids. Conjugate lipids. So, conjugate lipids are what? ये क्या हो गया? ये ऐसे स्लाइड ऐसे भाग कैसे गया था बीच में? चलो आ जाओ, वापस आ जाओ. Conjugate lipid is what? Conjugate lipid is the lipid के साथ कोई और present है. Conjugate, co-living भी say ना. Sir, we will go to a hostel and co-live with our roommates. So, conjugate lipid is what? Lipid is not akela. Lipid is not alone. Lipid is what? Having some non-lipid part. So, lipid plus non-lipid is making conjugate lipid. Okay? Lipid and yes, I got confused. Not confused, basically. Something happened with my tongue. So, lipid and non-lipid. Lipid and non-lipid. अगर होगा तो we are going to call it as conjugate lipid. Example what? Example is phospholipid. Phospholipid. Now guys, where this phospholipid is present? ये भी तो बता दो मुझे. Yes, Sakshi, it's an academy class. See? Yes. Okay. So lipid plus non-lipid is what? Lipid plus non-lipid is the phospholipid. Okay. Where it is present? It is present in the, खुद बताओ, तुम मैं सारी चीजें बता दूं? हाँ, सारी चीजें मैं ही बता दूं. Easy explanation, if I talk about the Campbell biology, it's a very good book. But for us, see, if we need to study something and if we teachers need to revise something, it's very good for us, but not for you, because you don't have to go into that high level. Okay, right? So stick to the NCERT. You can just refer that book for some explanation. If you're not able to understand something, just refer that then. Okay, but ऐसे देखा जाए तो NCERT is more than sufficient to you, for you. Lipid is with his wife. Yes, good. Lipid is with his wife. Okay, so phospholipid is present where? Plasma membrane. Plasma membrane, right? Now we have another one which is derived lipid, which is derived lipid. 1 second, bachcho. Just a second. Which is derived lipid. Derived lipid. Okay, just a second. I will connect my laptop to charger. Okay, 1 second. See, before the starting of the session, I had put everything, like I had put the charger in the plug, everything, but I forgotten to switch on it, and now my laptop was saying, "अरे अरे मैं मरने वाला हूँ, बचाओ मुझे." चलो, let's get back here. Derived lipid. Okay, so derived lipid is what? Derived lipid is not lipid. Derived lipid are not lipid. They are what? They are esters. They are esters. ये lipid नहीं है. They are esters, but properties are of like lipids. Behaves as lipids. Example is cholesterol. Example is cholesterol. Okay, cholesterol. Clear? अरे what I was saying, देखो साक्षी तुम्हारा कमेंट मुझे सब चीज भुला दिया. What I was saying, क्या पढ़ा रहा था मैं? Derived lipid is the cholesterol is the example of derived lipid. Okay, cholesterol is the example of the derived lipid. Clear? चलो, great. Now see, one more thing, two very simple lipids. One type of lipid is glycerol. Very simple lipids are glycerol and fatty acids. Okay, glycerol and fatty acids. See here, this glycerol and fatty acid makes what? This glycerol and fatty acids makes the simple lipid. Okay, this glycerol and fatty acids makes the simple lipids. So कैसे बनेगा ये? Hi Shai, you are back. Great. So glycerol and fatty acids are making the simple lipids. So see here, what is glycerol? What is glycerol, sir? So see, 3 carbon. Let's take the 3 carbon. Okay, and OH लगा दो यहाँ पे. OH, and here we will be having H and H. H for Hirsch. Very good. This is H and this is also H. Now what you do is that, if you need to make the glycerol, if you need to make the fatty acids, monoglyceride, triglyceride, ester bond, yes, kind of esterification. So what I'm going to do is that, see, I'm going to bring an ester. Okay, sorry, fatty acids. I'm going to bring a fatty acid. This is what? This is glycerol. This is glycerol. So मैं क्या लेकर आऊंगा? I'll bring a fatty acid. So fatty acid, C, double bond O, R, and here it will be what? It will be OH. Okay, then what I'm gonna do is that I'm just gonna react this. यहाँ से I'll remove the H. 1 second. Again बच्चों, देख रहे हो ये इरेजर बॉन्ड जो है काम ही नहीं कर रहा. इरेजर बॉन्ड सर, इरेजर टूल. See, so from this what I'm gonna do is that I'm gonna remove the H. So कैसा बॉन्ड बनेगा? See, how bond will be formed? Something like this. 1 second. So let us remove this H from here, and what is going to happen? Let's remove this OH from here. So the bond which we are gonna get is what, sir? The bond which we'll be getting is something like this, C double bond O and R. So if suppose, no Sakshi, it's fine. थोड़ा बहुत तो distraction होना चाहिए ना. Otherwise you will be like, hey, biomolecules पढ़ लिया, बहुत boring था lecture. Yes or no? Yes. Chalo. But that's a very good example, Sakshi. I'm gonna use it in every class now. And do you want, I don't think so you claim the copyright, right? ऐसे तो नहीं आ जाओगी सर copyright है इस example पे. Yes or no? ऐसे मत करना. So see, if I'm gonna add all the, if I'm gonna remove all the OH and I'm gonna add the fatty acids here, now this structure is what? This structure is half of the triglyceride. Clear? Clear to this structure is the triglyceride. Okay? याद हो जाएगा क्या? This is given in the NCERT बच्चों, so you have to learn it. You have to learn it. Clear? Great. चलो. Now we have conjugated lipids. Now we have conjugated lipids. Lipids will be completed. Enzymes I will do, and then the chapter will be over. Enzymes is a very small part. Okay? So don't worry. Conjugated lipid. Conjugated lipid. See, for you, I have launched the one-shot sessions, okay, everyone? So, everyone will be attending these sessions, and I promise you that within 1 week, we will complete the whole human physiology. हाँ, आधे बच्चे सोच रहे हैं 1 वीक में बायो खत्म कर दोगे क्या सर? 1 वीक में कैसे हो जाएगा बच्चों, बताओ? हाँ, मैं तो करा दूं, तुम कैसे करोगे? वैसे मैं भी नहीं करा पाऊंगा. चलो इधर आ जाओ. So one is what? Phospholipid. Phospholipid. So phospholipid का structure we have to remember. हल्का बहुत, थोड़ा बहुत you. Okay, Sakshi. No issues. So थोड़ा बहुत, yes. So थोड़ा बहुत phospholipid का structure you remember it, okay? But not very. ऐसा नहीं कि तुम्हें exam में बनाने आएगा. If in exam you have given the option, then you should identify that that is the phospholipid. For that sake, we are making these structures, okay? Yes or no? Great. Chalo. Now see here. We have this triglyceride, okay? Sorry. We have this triglyceride, yes or no? So in triglycerides, sir, what we saw was, this was O and here was the fatty acids and ester bond, yes or no? Yes. So in the second carbon also we are going to make it like this. But in third carbon, instead of this, what we are, acha, you are a JEE student. Sakshi. But you have taken biology, right? Is that so? Is that so? Guys, see, NEET wale bachcho dekho, JEE students are here and they are studying biology, but you guys are not studying, huh? Is that so? No, my all... Okay, Sakshi, bye. Chalo, let's come here. Let's come back here. So CO, see, instead of fatty acids, what I'm gonna put it here is that phosphoric acid. Phosphoric acid. यहाँ पे क्या लगा देंगे? Phosphoric acid लगा देंगे. जो बड़ा सा गंदा सा दिख रहा ना, ये P है तुम्हारे लिए. Okay? चलो. एंड इस फॉस्फोरिक एसिड में हम एक और ग्रुप लगा देंगे, कोलिन ग्रुप. एक और लगा देंगे कोलिन ग्रुप. देख रहे हो ये कितना बदमाश सा बच्चा है ये, हाँ? इतना सारा चीज लगा के बैठा हुआ, कोलिन ग्रुप, ये ग्रुप, वो ग्रुप. सो दिस इज द स्ट्रक्चर ऑफ phospholipid. This is the structure of phospholipid. So if it comes in exam, basically you should have a general idea that okay, this is the structure of phospholipid. Chances are less, but see, we don't know na this time what NTA is gonna do. No one knows. Okay, chalo, clear? This is the structure of phospholipid. Hi Anjali, how are you? वापस आ गए? No, okay. Is that so, Shakshi? Chalo. So next is glycolipids. Example of glycolipids are what? See, one was what? One was phospholipid. So phospholipid में lipid plus phosphorus. Phospholipid में क्या होगा? Lipid plus phosphorus. Okay. And if I talk about the another conjugate lipid, which is glycolipids. Glycolipids. So in glycolipids, what we are gonna have? We are gonna have lipids plus carbohydrates. We are gonna Sinchan is back. Yes, Sinchan is back. Lipid is gonna have what? Lipid is glycolipid is gonna have lipid plus carbohydrates. Okay, lipid plus carbohydrates. 1 second. 1 second. Who was the best friend of Sinchan? There was one character ना जिसका ऐसे निकलते रहता था नाक से. What was the name? Choline group. Choline group. Choline. अच्छा, I have not written it. 1 second. This group is known as choline group. CHOLINE. CHOLINE. Okay. Sakshi, because it was planned 23 hours ago. That's why. Don't worry. Don't worry. I'm not standing for 28 hours here. Otherwise, I would have. Right? Glycolipid is what? Glycolipid is lipid plus carbohydrates. Lipid plus carbohydrates. Clear? Next is lipoprotein. Next is lipoprotein. So lipoprotein is what? Lipid plus protein, right? Lipoprotein is what? Lipid plus protein. Clear? Any doubts till here? Batao. Any doubts till here? Anyone? No doubts? Any doubts anywhere? No doubts? Great. Okay. Yes? Chalo. Good. Very good. Very good, bachcho. So we have completed lipids. We have completed proteins. We have completed carbohydrates, right? Now it's time to complete enzyme and it will just take 10 to 15 minutes, okay? It's just time to complete enzyme. It will take 10 to 15 minutes, right? करवा दूं ना? Yes or no? Yes. See here. Let me talk about the enzyme. Let me talk about the enzyme now. So, enzyme are what, sir? Enzyme are catalyst. What kind of catalyst? Biocatalyst. Let's suppose that you are not able to do a certain reaction in your body. So, what will come? Enzyme will come and enzyme will say that आ जाओ मिलके हम reaction को complete करते हैं. So, body बोला कि reaction को complete करना मेरा aim नहीं है. Reaction को जल्दी से complete करना मेरा aim है. Clear to everyone? Yes? चलो. So, see, enzymes are what? They are acting as a biocatalyst. They are acting as a biocatalyst. Yes or no? Yes, sir. 1 second. 1 minute. क्या हो गया बच्चों? चलो, great. Kazma best friend है सिंचन का. Yes, Kazma. Yeah, I remembered. And there were so much beautiful cartoons, na? Like Bob the Builder and that octopus one. What was that? What I used to do is that I used to leave school to watch these type of cartoons. चलो आ जाओ वापस आ जाओ वरना बैठ के अभी सर वो वाले एपिसोड में ऐसे हुआ था याद है कि आपको वी विल स्टार्ट डिस्कसिंग दैट चलो कम हियर सो नेक्स्ट वी हैव दिस enzymes. So enzymes are what? Enzymes are biocatalysts. Now see, all the enzymes are what? They are proteins. All the enzymes are what? All enzymes are basically proteins. Okay, all enzymes are proteins. जितने भी enzymes है बच्चों सभी प्रोटीन है लेकिन it's biology. एक ना एक कोई नालायक तो निकल जाएगा. एक ना एक कोई नालायक तो ऑगी एंड द कॉकरोच. एक ना एक नालायक तो निकल जाएगा. यहाँ नालायक कौन है? यहाँ नालायक कौन है? राइबोजाइम. यहाँ नालायक कौन है? राइबोजाइम. एक्सेप्शन इज व्हाट? एक्सेप्शन इज राइबोजाइम. डोरेमोन. Yes, it was one of my favorite also. Ribozyme. And you know, do you know these ones? Karishma ka Karishma and Shararat, all those things. Have you watched these things? See, so RNA, this ribozyme is what? Ribozyme is an exception. It is not a protein. It is what? It is RNA. It is not a protein. It is RNA. Yes or no? Clear? Chalo. Now, let's focus on the enzyme, bachcho. Otherwise, when the question is gonna come, you will be like, ये टॉपिक मैंने पढ़ा नहीं क्योंकि इस टॉपिक के दौरान हम सारे लोग कार्टून्स के ऊपर डिस्कस कर रहे थे, यस और नो? चलो, so exception is what? Ribozyme. Exception is ribozyme. Now see here, enzyme is what? Enzyme is basically, see, enzyme is what? It is a very, arrogant kind of banda. Enzyme is what? Very arrogant. Very arrogant. Arrogant. Okay. How, sir? See, when we are gonna ask enzyme, कि भाई reaction करा दे, when we are gonna ask enzyme कि भाई start the reaction, enzyme will be like, I can't start the reaction. So enzyme never starts the reaction. Never starts the reaction. Starts the reaction, okay? He says that you start the reaction, then I will do my work. You start the reaction, then I will do the work. So enzyme is doing what? It is never starting the reaction. Rather it is making the reaction fast. Rather, it's making the reaction fast. Okay, it's like this. See, it's what? It's like this. Like you are going somewhere. Okay, you are going somewhere and you took an auto. Okay, you were about to took an auto rickshaw, but when you put your hand in your pocket and you realize that you don't have any money, then what? Then you were walking and suddenly there is a very good person who came and who provided a lift. So that person is what? That person is an enzyme. He is just helping you to go home fast. Okay, if it is a good person, then he will drop you at home. If it is a bad person, he will kidnap you and then ask your parents to give the firauti, right? So whenever you take a lift, check whether the person is good or not, or else lift nahi lo, theek hai? That is should be there. Chalo, let's move on. बच्चों. So see here we have one graph जो हमें सपनों में आके डराती है. Yes or no? This graph, sir, हमें सपनों में आके रोज डराया करती थी. अरे तुमको क्या हो गया? चलो, see, within 10 to 20 minutes we are gonna complete this. Don't worry. अब 10 to 20 minutes भी नहीं लगेगा बच्चों. See, when you will gonna revise this from the NCERT, na, I assure you that each and everything will be very clear. Each and everything will be very clear, bachcho. So see, what is happening on this x-axis, sorry, y-axis, I'm gonna take potential energy, and on this x-axis, I'm gonna take the progress of reaction, okay? Progress, or time I should say, progress of reaction, right? So see here, basically to make a product, what do we need? We need a substrate. We need a substrate, okay? We need a substrate. Now this substrate reacts with something and it forms a transition state. It forms a transition state, and from this transition state we get the product. So how, sir? Let's suppose that you are making a biryani. Let's suppose that you guys are making a biryani. How many of you like the biryani? How many of you love the biryani, guys? Okay, so you can order it for yourself, right? If you like this biryani, I'm not gonna give. ये मत सोचो कि मैं deliver कर पाऊंगा, बिल्कुल नहीं. When you guys crack the NEET, then I can maybe give you a party, but not now. Chalo. So if suppose you are making a biryani, like, so what you are going to do? First of all, you are going to boil the rice, then chicken, and then you will do. So if I come, like suppose you are making a biryani and the guest arrives early, तो ऐसा तो नहीं होता कि बस थोड़ा सा निकाल के दे देते बन गया. नहीं, बिरयानी बनने में टाइम लगता है. बीच में वो बिरयानी भी नहीं कहलाएगा, चावल भी नहीं कहलाएगा, something else only. So transition state is a state where the product is also not there, the reactant is also not there, something other is there, right? Now then slowly the transition state will get converted into the product, right? So see here, this is what? This is your substrate. This is your substrate here. Now this is the energy of the substrate. Yes, this is the energy of the substrate. Now what? This substrate is going to what? It is going to gain some energy. This substrate is going to gain some energy and it will go into transition state, right? And then what? From the transition state, it will make the product. So this one is what? This gap is the transition state, okay? This is also known as activation energy, EA. In chemistry, chemical kinetics, you have seen this, right? EA, activation energy. activation energy. This is what this is, the activation energy. Now, if you have used the enzyme, so enzyme will do what? It will lower the activation energy and finish the reaction fast. So this white is what? White is when we used the enzyme, and yellow was what? Without enzyme. Yellow was what? Without enzyme. It's just like if you are making the biryani, everything, all the masalas and all you are making, so it will take time. But if you buy the ready-made masala from the market, it won't take much time. Okay, so that ready-made masala is the enzyme here. Clear? Any doubts till here, bachcho? Any doubts till here? No doubts? Okay. Yes, yes. Now see, let's talk about the factors affecting enzyme. Let's talk about the factors affecting enzyme. See, if you're going to increase the temperature, so what is going to happen? First, it will increase the activity of enzyme. First, it will increase, but then it will decrease. Okay, then it will decrease. So let's suppose you are increasing the temperature. The temperature is 10 degree, let's suppose. Right now, the temperature is 0 degree, suppose, right now. So the enzyme was inactive. At 0 degree, that enzyme was inactive, just for an example. And at 10 degree, let's suppose, it became active and started working. At 20 degree, more active. At 30 degree, maximum active. So this will be known as what? Optimal temperature. This will be known as optimum temperature. Okay. But if further you increase the temperature, like you did it 40 degree, 50 degree and 60 degree, again the enzyme will become inactive. Again the enzyme will become inactive, right? So there is a line in NCERT that for every 10 degrees Celsius, increase in temperature, for every 10 degree increase in temperature, what happens, bachcho? Enzyme activity increases or doubles, but only up to a limit. ऐसा नहीं कि तुम 100 डिग्री कर दिए तो बस अब तो एंजाइम एकदम चलेगा, एकदम जनरेटर लग जाएगा उसमें, ऐसा नहीं होगा, राइट? वो भी बेचारा RIP हो जाएगा. Clear to everyone? Yes or no? Yes or no? Same thing happens with the pH also. Same thing happens with the pH. pH का भी same thing. Okay? Yes? So at some pH, it is what? It is working properly. But then agar you are increasing the pH, then again it will what? It will deteriorate. Yes, Shri Sai, denature. Again it will deteriorate. Clear? Chalo. So this is the graph again for the pH. So at some pH, lower pH, it will be inactive. There will be an optimal pH which is 7.8 pH. Okay, 7.8. If I talk about some enzyme like 7.8, maybe some enzyme is working at 7. So each and every enzyme in our body is having different optimal pH for working. Okay, सारे enzymes का अलग अलग pH होता है काम करने का, optimal pH. Maybe pancreatic enzyme 7.8 में वर्क कर रहा हो, maybe हमारा stomach का enzyme वर्क कर रहा हो acidic pH में, right? So this you have to figure it out, like each and every enzyme is having different optimal pH. Each and every enzyme have different optimum pH. Clear to everyone? Bachcho? Yes or no? Great. Now let's again move on and let's talk about the very bhyanak cheese. What bhyanak cheese, sir? The very bhyanak cheese is basically the Km constant. Yes or no? The Km constant. Acha, before that, let me talk about the active site. Bachcho. Just 10 minutes more. Okay? After that, the chapter will be completed. See, we have done biomolecules in so less hours. Like it's been 2 hours, 10 minutes more, we will complete. So active site is what basically? See, suppose that this is an enzyme. Okay? and in enzyme just like puzzle, let's suppose you are having a puzzle and उस puzzle में ऐसे खाली जगह होता है ना दूसरा कुछ लगाने के लिए, so that empty, yes, मैंने गलत लिखा क्या? For every, one second, yes, different, yes Anjali, yes, चलो ठीक लिखा हूं मैं, I thought I made a mistake. So active site is what? Active site in an enzyme is basically a curve like structure, a place where the substrate is gonna come, where the substrate is gonna attach, and what is going to happen? It is going to go into the transition state, and then again this substrate is going to be released from the enzyme, and it is now being called as product. Okay, this is what, this is the active site. Okay, a pocket like structure, okay, clear? Yes, great. Now see, the enzymes are never used in the reaction. Enzymes kabhi khatam nahi honge, yes or no? Like let's suppose you have used 100 enzymes, 100 molecules of enzymes. So at the starting of the reaction, if it was 100, so ending maybe it will be 100 only. Ending maybe it will be 100 only. Okay, enzymes never get consumed, never get consumed in the reaction. Okay, never get consumed in the reaction. Clear to everyone? Never get consumed in the reaction. Okay, now chalo अब हम आते हैं Km constant के बारे में. Okay, concentration. Let me now talk about the concentration of substrate. See what we have seen, sir. We have seen how pH is affecting the enzyme. We have seen what? We have seen how the temperature is affecting the enzyme. Now we are going to see how the concentration of substrate is going to affect the enzyme. See here, a very valid question, a very valid sense, guys, a very valid sense. See here, I'll ask you a question that if suppose there is 100 enzyme, okay, if suppose we have 100 enzyme, okay, right, now I have right now at time is equal to 0, let's suppose I am having 10 substrate, at time is equal to 0, I am having 10 substrate, so the graph will be something like this. Next what? At time is equal to 30 seconds, I am having let's suppose 20 substrate, so something like that, this I am doing. Okay, but ये क्या बन गया बच्चों, रुक जाओ, ये तो बहुत ही अलग सा ग्राफ बन गया. One second, let me make it again. Okay, let me make it again. Okay, the ones who are staying here, I'm gonna give you a gift. What gift, sir? I'm going to give you the question of biomolecules. Okay, I'm gonna give you the questions of biomolecules. Okay, chalo. Now see here. Yes, I'm gonna tell you, I'm gonna help you with this. Okay, don't worry. See, so what is happening? You are having the 100 enzymes. Okay, and at time t is equal to 0, you were having 10 substrate. Then what you have done? At time t is equal to 0, at time t is equal to 20, let's suppose you are having 30 substrates. So slowly what you are doing? Slowly you are increasing the number of substrate. Okay, and you have seen that the speed of the reaction is also increased. The speed of reaction is also increased. But sir, now I have substrate which is 110, but still the speed of reaction is not increasing. Why, sir? Because we had 100 enzymes. Yes or no? We had 100 enzymes, 100 active sites. Now you have 110 substrate. All the 100 enzymes are working. 10 substrates don't have any enzyme. So now what is going to happen? The speed of the reaction will become constant. The speed of the reaction will become constant. Okay, clear to everyone? Yes or no? Yes or no? Anjali, right now so we don't have as such, our official Telegram group is not there. When it will be made, I'll tell you. Okay, I'll inform you. Okay, great. Now see. So this is what, did you understood, Srishai? Did you understood this? Did you understood, bachcho? What you can do is, Anjali, download the Unacademy app and follow the community there. Follow my community there. Okay, You can follow my community there. Unacademy app download karo, search Harsh Kumar, and then you join my community. Now see, bachcho, check here, check here. So what is happening? At this point, the velocity is constant, and that's why the graph is like this. Clear to everyone? This is the point where the velocity is maximum. Now, kitna bhi tum kya kar lo, kitna bhi tum substrate ka concentration बढ़ा लो, now the velocity is not going to be increased. Clear to everyone? Okay, 1 second, bachcho, 1 second. Chalo. I will make this graph little thoda sa modify kar lu. So this is the point where Vmax is happening. Okay, this is the point where Vmax is happening. Now see, sir, there is Km constant. There is Km constant जो हमें कभी समझ में नहीं आता. देखो, this is what, this is Km constant. What is Km constant? What is Km constant? See, let's suppose that there will be one point where the velocity is, let's suppose maximum velocity is 10. Let's suppose maximum velocity is 10. Okay, Vmax is 10. Okay, there might, Vmax is 10 at 100 substrates. When you have 100 substrates, then Vmax is 10. Okay, now there might be a point where Vmax will be half, maybe 5. Yes or no? ध्यान से समझना. There will be a point where Vmax will be what? Vmax will be half, right? So Vmax is what? 5 here. But what is the amount of substrate here? Maybe the amount of substrate is 30. So whatever the amount of substrate is there, whatever the concentration of substrate is there, to reach the Vmax is known as Km constant. Whatever the amount of substrate is there to reach the half of the maximum velocity, that is known as what? That is known as Km constant. Yes or no? Clear? So I'm going to write it for you all. What is Km constant? Concentration at which? Concentration of substrate. Sorry. I'll write here. Km is what? Km is concentration of substrate at which the velocity is half of the maximum velocity. Is it clear to everyone? Is this thing clear to everyone? Yes or no? Chalo. Now though I don't think so you would have any doubts, right? Now see, let's talk about the enzyme inhibition. Now what we are having now, we are having competition here. Last topic is enzyme inhibition, and then we have the cofactors and all. Then it will be over. Okay, then it will be over. Chalo, just 10 more minutes. Okay, enzyme inhibition. I know from the past 15 to 20 minutes I am saying 10 minutes, but this 10 to 20 minutes will be worth it, guys. These time will be worth it. Okay, chalo. At the few time I was having 10 to 11 or 12 live students, but see, now we are just 4 of us. So this is, like, you 4 will have this thing in your mind that yes, we have completed this chapter, and जो सुकून मिलेगा ना ये सोच के कि हमारा एक चैप्टर कंप्लीट हो गया, that will be very, very good for you. Okay, चलो. Now let's talk about the enzyme inhibition. सुन रहे हो? Now, so 2 types of enzyme inhibition is there. One is competitive. See here, I'm going to give you an example, a very good example. If suppose I ask you, Shri, Shai, Anjali, Bhavik, Shivesh, if I ask you everyone, so what you are guys doing? You are preparing for NEET. Yes or no? And suppose this is a medical seat. Yes or no? This is a medical seat. And I asked you to do what? I asked you to play musical chairs around it. Okay. So let's suppose Bhavik, what it did? Bhavik said that, ki, sir, I will reproduce myself and I'll make copies of myself. Let's suppose 3 Bhavik आ गया, 3 Bhavik आ गया. एक श्रीशाई है, okay, and one is Shivesh. So probability of getting medical seat for Bhavik will be what? It will be increased. Yes or no? Yes, because Bhavik कितना आ गया? 3 Bhavik आ गया. In a similar manner, guys, see, this is what? This is an enzyme. This is an enzyme, okay? And this is its active site. This is its active site, okay? This is its active site. So in competitive inhibition, what happens na? जो inhibitor है, it is what? It is just like substrate. It is just like substrate, okay? But here what I have done is that I have increased the concentration of substrate. Increased the concentration of substrate. So क्या होगा? Substrate का active site में जाने का probability बढ़ जाएगा. Yes or no? Yes. Substrate का active site में जाने का probability बढ़ जाएगा. Yes or no? So reaction will to complete. Yes or no? Reaction will to complete, but late से होगा. Reaction will complete, but late से होगा. See here. This is what? This is the again Vmax graph. So here what is happening? Earlier the graph was like this. Earlier the graph was like this. Now the graph will be something like this. The graph will be something like this. So what happened here, sir? What happened? The value of Vmax is increased. Sorry, the value of Km constant is increased. Yes or no? This blue line represents what? Competitive inhibitor, and this white line was what? This white line was normal. So see, normally Km was what? Km was less, and in terms of competitive inhibitor, what is happening? The Km is more. Okay. So in competitive heat, hi Karan, how are you? In competitive inhibitor, what's happening, sir? In competitive inhibitor, the Km constant is what? The Km constant is increased. Yes or no? Yes or no? Is it clear? See, Vmax तो same है. Vmax को तो कुछ नहीं किया हमने. Vmax को तो छेड़ा भी नहीं. Clear to everyone? Clear to everyone? Yes or no? Yes or no? And in non-competitive inhibitor, example of competitive inhibitors are malonate, inhibition of succinate dehydrogenase by malonate. मैं example लिख दे रहा हूँ यहाँ पे. Succinate dehydrogenase by malonate. Okay, by malonate. Okay, clear to everyone? Will this inhibitor enzyme? No. Inhibitor will not form the product. Inhibitor will not form the product. नहीं बनाएगा वो product. वो बस चिपक जाता है. वो चाहता है कि product बनाने ना दे. खुद कुछ नहीं करेगा वो. Okay, clear? Very good. चलो. Next we are okay. Anjali is also here. Sinchan is also here. So next we are what? We are having non-competitive inhibitor. Non-competitive, clear ना? Great. Non-competitive inhibitor, ये भी देख लो. Non-competitive inhibitor, clear? So see here, what is non-competitive inhibitor, sir? Non-competitive inhibitor is very chalak. It is very chalak. ये क्या करता है ना? ये negative friends के जैसा होता है. So see, this is the active site in the enzyme, or in the enzyme there is some other sites also which is not active site. Okay, which are not active site. Let's suppose this. So non-competitive inhibitor will do what? It will come here and it will say, पता है साईं अच्छा नहीं है, वो ऐसे करता, वो तुम्हारे बारे में गंदा बात बोल रहा था, उससे दोस्ती तोड़ दो. तो ये क्या करेगा? ये अपना एक्टिव साइट का शेप ही चेंज कर लेगा. ये एक्टिव साइट का शेप ही चेंज कर लेगा. अब तुम कुछ भी कर लो, यहाँ पे कौन नहीं आ पाएगा? सबस्ट्रेट नहीं जुड़ पाएगा. यस और नो? सबस्ट्रेट नहीं जुड़ पाएगा. क्लियर टू एवरीवन? क्लियर? यस श्रीशाई, वेरी गुड. यस, एग्जांपल इज व्हाट? साइनाइड पॉइजनिंग. यस और नो? साइनाइड binds with cytochrome oxidase. Cyanide binds with cytochrome oxidase. Clear? Oxidase, right? चलो, ग्रेट. नाउ नॉन कॉम्पिटिटिव ग्राफ भी देख ले क्या? थोड़ा सा नॉन कॉम्पिटिटिव ग्राफ भी देख ले क्या? सी, सो दिस इज व्हाट? दिस इज द नॉन कॉम्पिटिटिव ग्राफ. वन सेकेंड बच्चों. दिस इज द नॉन कॉम्पिटिटिव ग्राफ. and here before the graph, before the competition, what's happening, it is like this, it is like this, okay. But after the competition, what's happening, Vmax is decreased. Now see, amount of enzyme is less. अब क्या हुआ, एंजाइम का अमाउंट कम हो गया ना तुम्हारे पास, तो अमाउंट ऑफ, सांस नहीं ले पाते, यस, कौन नहीं ले पा रहा, कौन नहीं ले पाता, अच्छा फाइनाइट वाला में, यस यस यस, भाविक, अंजलि, श्रीशाई, शिवेश, सो गए क्या, सो गए क्या शिवेश, चलो चलो चलो, सी सो एंजाइम क्या हो जाता है, एंजाइम का अमाउंट डिक्रीज हो गया, कैसे सर, ये खराब हो गया ना, कुछ तो एंजाइम खराब हो गया तुम्हारे पास, तो एंजाइम का अमाउंट जब डिक्रीज हो जाएगा तो तुम्हारे रिएक्शन क्या हो जाएगा, रिएक्शन का स्पीड भी डिक्रीज हो जाएगा, राइट, yes or no, but there will be no change in km constant, km constant will be not changed, right, km constant will not be changed, okay, चलो, yes yes, cyanide वाला, km constant will remain constant, and कितना अच्छा लाइन आ गया ना, km constant will remain constant sir, चलो one second, and Vmax will be what, Vmax will be decreased. ये ग्राफ इतना टेढ़ा क्यों बन गया बच्चों, बताओ, कोई बात नहीं, तुम अपने कॉपीज में बना रहे होगे ना, तो वो कर लेना, सीधा बना लेना इसको, Vmax what 2, okay, Vmax 2, okay, clear, चलो great. Now just the last topic बच्चों, just the last topic, and then we will be finishing the biomolecules today, just the last topic बच्चों, चलो, एंजाइम के साथ साथ भाई होता है, एंजाइम का क्या होता है, भाई, एंजाइम बोलता है मैं अकेला काम नहीं कर सकता, मुझे कितना काम करना पड़ता है, याद है ना, एंजाइम क्या था, एरोगेंट था, बहुत एरोगेंट था, तो एंजाइम बोलता है मैं अकेला काम नहीं कर सकता, तो मुझे क्या चाहिए, मुझे चाहिए कोफैक्टर्स, मुझे क्या चाहिए, मुझे कोफैक्टर्स चाहिए, रुक जाओ रुक जाओ, माय लैपटॉप इज लाइक, थोड़ा सांस लेने दो, माय लैपटॉप इज लाइक, थोड़ा सांस लेने दो, sometimes I think that if suppose my dog is able to learn things, he would be a god of biology, right, he would be a god of biology, उसको नीट दिलवा देता मैं, है ना चलो, now see here, let's have cofactors, let's have cofactors, what is this cofactors sir, see, enzyme is what, enzyme can't do work alone, so enzyme need what, enzyme need something, enzyme need something, so if suppose that I am talking about the holo enzyme, if I am talking about, चलो होलो एंजाइम अभी इंट्रोड्यूस नहीं कराता, तो पहले समझा देता हूँ, फिर शीशा क्लियर है ना, वो ग्राफ क्लियर है ना, कोई डाउट तो नहीं है उसमें, इज इट क्लियर, कोई डाउट तो नहीं है शीशा उस पे, चलो मान लो ये तुम्हारे पास एंजाइम है, क्या है ये, एंजाइम, एंजाइम, ओके, नाउ दिस एंजाइम विल बी मेड अप ऑफ व्हाट, इट विल मेड अप ऑफ प्रोटीन पार्ट, हमने बोला था प्रोटीन से तो बना हुआ होता है एंजाइम, तो प्रोटीन पार्ट, और साथ में कौन इसको चाहिए, कोफैक्टर, कौन चाहिए इसको, कोफैक्टर, राइट, जो इसका थोड़ा बहुत काम कर दे, सो सी, इस प्रोटीन और कोफैक्टर को हम क्या बोल देंगे, एपो एंजाइम, नाम थोड़ा सा चेंज हो गया, सॉरी होलो एंजाइम, नाम थोड़ा सा चेंज हो गया, होलो एंजाइम, है एंजाइम ही, है एंजाइम ही, बट ये पूरा एंजाइम है, अब प्रोटीन वाले पार्ट को क्या बोलेंगे, एपो एंजाइम, एपो एंजाइम, and this cofactor wala part will be known as non-protein part, okay, this is non-protein part, clear, now we have to see that which one is which, okay, we have to see that which one is, sorry, types of cofactor, kya bol raha hu main, types of cofactor, next we have to see types of cofactor, okay, types of cofactor, now see here, what are the different types of cofactor, one is the prosthetic group, prosthetic group, see, prosthetics are what, prosthetics you might have heard that these are those kind of equipments which are used, Vmax change होगा तो km constant भी change करना होगा ना, नहीं, Vmax, see, km constant क्या बोल रहा है, km constant वो constant था कि मान लो Vmax तुम्हारा है, Vmax नॉर्मली देखो, नॉर्मली Vmax तो यहां से चेंज हुआ ना, Vmax तो यहां से चेंज होगा, इधर से कहां गया वो ग्राफ, Vmax तो इधर से चेंज होगा बच्चों। देखो यहां तक तो चीजें सेम ही चल रही थी, मैक्सिमम वेलोसिटी, अरे ये कहां गया वो वाला ग्राफ, 1 सेकेंड, 1 सेकेंड, वो ग्राफ कहां चला गया। अच्छा मैं ऐसे समझाता हूँ, यहाँ पे क्या हो रहा था, यहाँ पे ये ऐसे था और ये ऐसे था, राइट। सो सी हियर what is happening, this part is changing, this part the maximum velocity is being decreased, right, but यहाँ तक तो सब चीज ठीक ही चल रहा था, यहाँ तक तो सब चीज ठीक चल रहा था, that's why km is not changed, okay, clear, yes। ये फिर से वो होने लग गया क्या, लैग होने लग गया क्या बच्चों, बताओ, let me know in the comment section। see, ओके चलो चलो ठीक है, अब ठीक है, अब ठीक है, अब ठीक है, चलो मैं फिर से समझाने की कोशिश करता हूं, मैं फिर से मैं फिर से समझाने की कोशिश करता हूं, अभी साक्षी का एग्जांपल लेंगे हम लोग, ठीक है। आई विल ट्राई टू मेक यू अंडरस्टैंड वंस अगेन। सी व्हाट इज हैपनिंग, बेसिकली km constant depends on the active site, Sri, okay, km constant depends on the active site, so non competitive inhibitor ना एक्टिव साइट में तो कोई चेंजेस नहीं आ रहा, एक्टिव साइट तो उतना ही है, तो अगर मान लो 50 एंजाइम 50 सबस्ट्रेट चाहिए हाफ वेलोसिटी को अचीव करने के लिए, तो वो आएंगे, देयर इज नो चेंज इन एक्टिव साइट, दैट्स व्हाई km कांस्टेंट इज नॉट चेंज्ड, ओके, क्लियर, क्लियर, ओके ग्रेट। चलो लड़ाई मत करो अब तुम दोनों, ऐसे तलवारें ना निकाल लेना तुम लोग। चलो लेट्स टॉक अबाउट दी टाइप्स ऑफ कोफैक्टर, टाइप्स ऑफ कोफैक्टर। सो प्रोस्थेटिक ग्रुप इज व्हाट, prosthetic is what, suppose that there is a person who lost his leg, so what we do is that prosthetics लगा देते हैं, and that prosthetic is what, it is there for his whole life, yes or no। hi Shruti, how are you, नमस्ते श्रुति, नहीं मुझे खुश रहो बोलना चाहिए ना, खुश रहो श्रुति चलो, so prosthetic group is what, prosthetic group is always attached, okay, always present, कभी छोड़ के नहीं जाएगा, कभी छोड़ के नहीं जाएगा, always attached, okay, always present, yes or no, always present, clear, right, and example is what, example is hemoglobin, hem part is what, sorry, not I'm sorry, hem part is prosthetic group, hem part is prosthetic group of किसका, नॉट हेमोग्लोबिन बच्चों, peroxidase catalase का, peroxidase catalase enzyme, okay, catalase enzyme। now next is what, next is what happened बच्चों, तुम हंसने क्यों लग गए, बताओ, next is what, coenzyme, next is what, coenzyme is basically what, साथ में, coenzyme is basically what, साथ में कभी कभी रहेंगे, what happened, साथ में कभी कभी रहेंगे, क्लियर, coenzyme is साथ में कभी कभी रहेंगे, हमेशा नहीं रहेंगे, मतलब जब जरूरत पड़ेगा ना तब आ जाएंगे, पैसे चाहिए तुम्हें, अरे भाई तू तो मेरा दोस्त है, मेरा जिगरी यार है, उसे पैसे चाहिए होगा तो मैं बोलूंगा जा, तो कुछ टाइम पे रहेंगे, कभी रहेंगे कभी नहीं रहेंगे। सो binds with apoenzyme, binds with apoenzyme loosely, okay, loosely binds with apoenzyme, clear, right। example is what, vitamins, example is what, vitamins are essential components, vitamins are essential components of coenzymes, okay, of some coenzymes। और क्या, जैसे कि एग्जाम्पल NAD, जैसे कि एग्जाम्पल क्या हो जाएगा, यस श्रुति, टेम्पोररी, यस वेरी गुड, NAD, फॉर एग्जाम्पल NAD एंड एक और होता है NADPH, किसके साथ बाइंड करता, B3, विटामिन B3 के साथ, ओके, विटामिन B3 के साथ, जिसको शायद नियासिन बोलते हैं, जिसको शायद नियासिन बोलते हैं, आई एम नॉट वेरी मच श्योर, बट इसको शायद नियासिन ही बोलते हैं. क्लियर? ओके, चलो नेक्स्ट वी हैव व्हाट, मेटल आयन. थर्ड चीज क्या है, मेटल आयन. थर्ड चीज क्या, मेटल आयन. अच्छा, ये दोनों चीजें क्या है, बोथ ऑफ देम आर ऑर्गेनिक, बोथ ऑफ देम आर organic. This is organic बच्चों, and coenzyme is also organic. Okay, this is organic and coenzyme is also organic, right? But this is what, this is inorganic, this is inorganic. And if I talk about the metal ion, so it is what, it forms the coordination bond. Right now I am taking class from my home, Sakshi. Forms the coordination bond. You know this chapter, oh my god, when I was in class 12th, this chapter was like, bhai kya hai ye? Forms the coordination bond. किसके साथ? With the side chain of active site and substrate. Okay, active site and substrate. Clear? Example will be what? Example will be zinc is a cofactor for carboxypeptidase. Carboxypeptidase. Clear to everyone, बच्चों? Yes or no? बोलो. स्वाहा हुआ कि स्वाहा हो गए? Chapter स्वाहा हुआ कि स्वाहा हो गए? Just one more slide and the chapter will be over. Just one more slide and the chapter will be over. Classification of enzyme. Classification of enzyme. Okay, classification of enzyme. See here, one is what? One is ligase. Yes, zinc. One second. One is what? Ligase. Okay, ligase. Oh, acha. Please, I am so sorry, Sakshi. I'll give you a guess. Okay, I'll give you a guess. A place which is hometown of one of the greatest cricketer. Should I give you the poll on this? But sir, I can't give the poll in YouTube. Okay, chalo. So ligase is what? Ligase enzyme. It is an enzyme which helps into जोड़ने का काम करता. Okay, which helps to ligate. So let's suppose this is one molecule and this is one molecule. So what it is going to do, it is going to create a bond between them. This is the work of ligase enzyme, right? This is the work of ligase enzyme. Second enzyme is isomerase. Yes. Isomerase. So what is this? Isomerase is basically what? Isomerase enzyme do what? It changes optical active को geometrical active में, like interconversion of isomers. Okay? ज्यादा अभी मैं तुम लोगों को ऑप्टिकल आइसोमर, ज्योमेट्रिकल आइसोमर बोलूंगा, तुम क्वेश्चन पूछ जाओगे, मैं ऐसे खड़ा हो जाऊंगा शांति से. सो आई शुड से conversion between different isomers. ये एंजाइम याद करो ना, ग्लूकोज 6 फ्रक्टेज. yes, ग्लूकोज 6 एंड फॉस्फेट एंड फ्रक्टोज 6 फॉस्फेट में ये एंजाइम यूज होता था क्या? यस और नो? होता था क्या? किस में होता था? नहीं सर, होता था क्या? चलो, कन्वर्जन बिटवीन different types of isomerase. Isomers लिखता हूं. Third is what? Third is the lyase. Okay, ligase का भाई lyase. So भाई हमेशा बदमाश होता, right? भाई क्या होता है? दो भाई तुम देखोगे, if suppose I am talking about two brothers in the same home, so one brother is very good and the other one is right, like he will destroy all those things. So ligase is gonna make things, and lyase is what? Lyase is gonna break the things. So what it is gonna do, suppose this is A and B, suppose this is A and B, so what is going to do is that, one second, it is gonna break them, okay. So this is lyase, exactly. Yes. Oh, I am so sorry बच्चों, I am so sorry. Yes. This is not present. Okay, चलो मैं पढ़ा दिया तो पढ़ लो, चलो दो ही और है. Hydroxylase. We have the whole thing na, like in a flow we teach also. Just 1 second. Hydroxylase. So this is what? This is gonna use water. MBBS में काम आएगा तुम लोगों को. चलो, this is gonna use water. With the help of water, it is what? It is breaking. With the help of water, it is breaking. So AH and BOH. So this is the hydroxylase. Okay. Next is transferase. So this enzyme is going to have what? See, if suppose A is attached with OH, so what is gonna do? ये क्या करेगा? A को अकेला कर देगा, B के साथ OH लगा देगा. ठीक है, धोखा देता है ये basically. Next is oxidoreductase. And this is what? A plus H. Let's suppose, 1 second. One second. So this is oxidoregistered is what? It is gonna oxidize. Let's suppose we have A के साथ हाइड्रोजन लगा देते, राइट? A के साथ हाइड्रोजन लगा देते, तो यहाँ पे क्या हो जाएगा? मैंने क्या किया? A से हाइड्रोजन हटा दिया और B को हाइड्रोजन लगा दिया. So इसका ऑक्सीडेशन हो गया, इसका रिडक्शन हो गया. क्लियर? चलो, ग्रेट. So this, now see here, there are some points which are primary metabolites and secondary metabolites, right? So primary metabolites and secondary metabolites का तुम्हें टेबल याद करना है NCERT से. Primary metabolites are those of which we know the function. Right? But secondary metabolites are those which we do not know the function. Okay? Do not know the function. Clear? Clear to everyone? Primary metabolites, we know the function. Secondary metabolites do not know the function. And you have to read it from NCERT. For this. Okay? So, almost I have covered all the important topics in this session. And if this session is full package, you just go and revise it from NCERT. If possible, revise it today only. And you will feel that we have completed a lot of things and lots of concepts. Okay, everyone? Okay. Tomorrow we are going to have same timing. We are going to have human physiology. Tomorrow we are going to have human physiology at 6 o'clock. 1 second. Which chapter I'm going to have? Maybe. Do you take classes? Yes. Every day, Shruti. Every day. Sorry, every day. And which subject? Biology. Shruti, biology at 6 p.m. every day. बच्चों मैं एक बार वो स्केड्यूल चेक करके बताता हूँ नेक्स्ट कौन सा क्लास है. 1 सेकेंड. बच्चों, आई हैव रिटन द स्केड्यूल एंड मैं ही भूल गया. 1 सेकेंड बच्चों, मैं बता रहा हूँ. Tomorrow is 22nd, right? So tomorrow we are going to complete circulatory system tomorrow. Okay, yesterday we completed breathing system or what? Breathing and exchange. Tomorrow we are going to complete the circulatory system. Okay, everyone? Okay. Sakshi, yes. G was glutamate. Bhavik, I'll see and let you know. Okay, I'll add it in the comment section. Yes, you're not a bio student. I'll add in the, acha, afsos. I will add it in the not अफसोस section guys, comment section. Okay, ठीक है भाविक? Okay, चलो 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 अब मैं जाऊं. It was a wonderful class guys. I literally enjoyed it. I hope so that you all also liked my class. So if you liked my class, leave a feedback in the comment section so that we all can improve as a channel whole. Okay, so till then, bye bye everyone. See you in the next class. Yes, human physiology tomorrow at 6 p.m.